Task Force and Microbiome Special Interest Group. More on this a bit later on. My name is Voki Odwaran, and it's my great pleasure to welcome you today. So welcome, Akwaba, Karibu, and apologies for the others I missed out on. As always, the aim is to showcase microbiome studies across the African continent. And this year we tried, keyword tried, to focus on health, ecosystems, and innovations. But we also wanted to make sure that as many studies as possible got some airtime. And so we do have quite a mixed bag for you today. So please sit back and enjoy. I particularly like that what you will see today, although not all-encompassing of the work going on the continent, does show the different stages and the different levels of research going on. You might not see some of the bigger projects you've heard about, but that's the joy in all of this, so that even all the other projects like doing smaller work, hoping to do some work, just getting started, gets some airtime as well, get to share what you're working on. So you can see, you might be able to see some of the varying capabilities across the different projects. So the format for today is that we have 3 main sessions and a joint keynote presentation that I am so looking forward to. The 3 sessions comprise both invited guest speakers and recorded presentations from selected abstract submissions. There will be 2 Q&A sessions during each session, one right after the invited guest speaker's presentation, and a joint live Q&A session for all the recorded presentations. Please use the chat and the Q&A option to ask your questions as we go along. We hope to keep the time as much as possible. And this chat functions in terms of interacting will help us do that. So we have about a 15-minute break after the second session, during which you can grab a cup of tea or coffee, stretch your legs, just check up your emails. We'll also be sharing a couple of poster presentations during that break as well. We'll also have a chance to just network and chat, maybe some breakout rooms. We'll see how it goes. If you'd want to just chat with a few people and just connect. The posters will have QR codes, so even if you feel like going through them very quickly, you can just scan them and have a look at them afterwards. Like I said, we'll have an incredible joint keynote presentation at the end of the event, so please do not rush off. And just before the keynote, we'll be sharing the vision of an African Microbiome Consortium. Really excited about this, and I hope you engage. You may have seen or not the QR code to a Google form where we're hoping to collate different studies going on the continent in different planning stages. We encourage you to fill that in as well. We're also happy to just get an email from you just to tell us, well, I'm not doing this just yet, but this is my prospective study. Let us know. All of this will go a long way in as we make tangible strides towards this vision of an African Microbiome Consortium. Tino Muzambi, his emails, probably you've seen it several times in the several communications we bombarded you with in the last few minutes and the last few days. My email as well is available. We're also happy to pop this in the chat if this might help. I would also like to especially thank all the moderators of the day in advance, an incredible organizing team, including Professor Morenike Upong, Dr. Brian Cullen, Prof. Lamek Wapaga, Professor Joan Pasmore, Dr. Edward Wapande, Dr. Aditola Louis Jacks, Professor Sumaya Kwedi, and the entire H3Africa Coordinating Center, with special shout outs to Dr. Francis Agama and Tina Muzambi. Really excited about today, and just to enjoy me to sit back, enjoy, engage, and have a fantastic event. Also, I'll be doubling in as the first moderator of the session this morning. We had a bit of a last-minute emergency, so I had to step in. So our first presenter of the day, I'm really excited about this. We've been trying to get a hold of him, and we're able to do that this time. Is from the University of Nairobi, Kenya, and a person of Professor Moses Obimbo Madadi. Professor Moses Obimbo Madadi is a Science Leadership Fellow. He's a clinician scientist and associate professor who is passionate about promoting learning, research, teaching, and clinical practice. He's using modern molecular tools in placenta biology, microbiome and obstetrics and gynecology research, such as laser capture, microdissection, and several multi-omics approaches, as well as artificial intelligence. He's keen on strengthening local and regional institutions, and today will be giving us a status update on the vaginal microbiome and metabolomics research in the Kenyan population. Please go ahead, sir.
Thank you very much, Vok, and thank you, colleagues here. It's a pleasure to present to you progress of my work on vaginal microbiome. I think if I'm allowed to share my screen, then I'll go ahead and do that. Yeah, thanks. I've seen I'm allowed to do that. So I'll share my screen. As Vok has indicated, professor at the University of Nairobi, and I'm doing work on vaginal microbiome and metabolomic profiles in adverse pregnancy outcomes in a Kenyan population. So I'll go ahead and make this presentation by just simply giving a background of this. As we all know, that Africa is disproportionately impacted the poor gestational and neonatal outcomes. And this, as a result, we have many pregnancies that end up in stillbirth, preterm birth, intrauterine growth restriction, and others postpartum like chorioamnionitis or endometritis and so forth. So this high burden of asymptomatic vaginal infection also affects and has direct correlation with the adverse pregnancy outcomes. We know very well that the microbiome, at least from the recent evidence, plays an important role in orchestrating both maternal and neonatal health, and it's therefore important to know the role it plays in pregnancy outcomes. We know that in the vagina, we have the microbiome, and the main component of the microbiome is the lactobacilli, which maintains and promotes the normal vaginal environment, and it does this through several mechanisms. The first is just maintaining the lower pH of the vagina, and then this bacteria also competes with the other offending bacteria for the nutrition, and also anchor on the adhesion sites of the bacteria, that therefore making it difficult for the offending bacteria to cause infections. They also do this through immunomodulation, and sometimes they also do this through release of bioactive substances or compounds that maintain the vaginal microenvironment, and that will be the focus of my study. We also know that the composition of this microbiome can vary from time to time. For example, in prepuberty, during the reproductive age group, and postmenopausal. And of course, during pregnancy, we might find different composition of vaginal microbiome. And this impacts the pregnancy outcomes. It's also been known that during pregnancy, the diversity of vaginal microbiome is reduced, and this makes it more vulnerable to changes, especially to infections. So the reproductive tract infections, as we know, is a very important clinical entity, but also a very important public health matter, because it does not only cause disease in the affected woman, but also affects their psychological state, presenting, things that will not be desirable during this pregnancy. So there is positive data that tries to relate the relationship between reproductive tract infection and the pathophysiology of adverse pregnancy outcomes, and how this also alters the vaginal microenvironment, including the vaginal microbiome and vaginal metabolomics. We also not have sufficient tools that can be able to identify pregnancies that are at risk, so that interventions can be put in good time. Recent studies as recent as 2024, 2021 have shown that the vaginal microbiome not only affects the maternal health, but also affects the health of the offspring. And in studies done in mice model showed that this microbiome is very important in the development of various systems, the neurodevelopment, respiratory system development, cardiovascular system development, and has given rise to the concept called the vaginal baptism during birth, encouraging vaginal birth as opposed to cesarean deliveries. So what's the need? We know that identification of the vaginal microbiome population and metabolomic is very important in relation to the pregnancy outcomes. And being able to do this would be able to help us identify which pregnancies are at risk of adverse pregnancy outcomes and develop tools that are relevant for either making the screening or diagnosis of these pregnancies in early stages. This has potential of changing our antenatal care practice so that instead of just doing the HIV, hepatitis B, VDRL tests and so forth, the ones that we do during pregnancy, we could potentially include a panel for testing reproductive tract infections as part of the antenatal care practice. We also have very minimal therapies that are targeted towards treatment of reproductive tract infections. And we hope through this kind of studies, we should be able to develop pathways that can develop live-based therapeutics to treat vaginal infections. Thus, this is a very important element that we need to be able to study in our setting to address the high level of adverse pregnancy outcomes that we have within our population. So the aim of our study is to first of all, analyze the vaginal microbial communities using the next generation sequencing. And the second one is to be able to identify the metabolomic profiles that are both predictive and diagnostic of various pregnancy, adverse pregnancy outcomes. The third one is to be able to correlate the metabolomic and the microbial data to the clinical characteristics. And now using all this information is to develop an AI-assisted predictive model that can be able to predict pregnancies that are at risk of adverse pregnancy outcomes. So we are able to carry out our studies in about 6 different hospitals in the Nairobi and out of Nairobi. And we've been able so far to collect specimens and samples from about 1,509 women. And the kind of characteristic of women we collected data from were women with who had normal term pregnancy, those with miscarriages, those with preterm birth, stillbirth, and IUGR. And what we did is that we got these women in the first trimester. We enrolled them in the first trimester of the pregnancies, and we were able to follow them and get these specimens from the first trimester pregnancy, second trimester, and those who were able to get to the third trimester, we were also able to collect the samples. And the samples we collected included the vaginal swabs, which we prepared in 3 different sets. We're also able to get blood, Baficord. We're able to get the plasma and able to get the serum samples for biobanking. We're also able to get urine samples and saliva samples for biobanking and for the future research questions. In a few patients, we're able to also collect stool samples and also the placental samples for further analysis, and we're able to put them in our biobank. So in terms of preliminary results, I'll just present a bit of the clinical results that we've had so far. Out of the 1,509 patients, some we had incomplete data. So the age distribution is as shown, and the median age was about 27 years. Most of the women who participated in this study were married. A few were single. Then most of the women had attained at least secondary education and tertiary education. Just a few had primary education. So by and large, most of these women were educated. In terms of employment status, we had just very few women who were in formal employment. Quite a number were not having any employment, so most of them were housewives. We also did blood grouping. We collected blood grouping as part of, we didn't test, but we had this as a part of our panel during the ANC investigations. And this was a distribution of the blood groups, and majority of our patients were blood group O positive, and followed by the A positive and B positive blood groups. So in terms of use of alcohol, most of our patients did not use any alcohol as indicated on this data set. And in terms of smoking, also most of our patients did not engage in any smoking activities. Our patients, most of them were healthy and did not have any chronic illnesses. Just a few had chronic illnesses like hypertension, pre-existing hypertension, or pre-existing diabetes mellitus. So in terms of pregnancy outcomes, and this was very interesting for our study, is that we had about 42 women with intrauterine growth restriction. We had about 121 women with miscarriages, 90 women with preterm birth. This is a low figure compared to what ordinarily you'll get in a normal population, giving about a rate of 8 to 9%. Then we had about 65 women with stillbirth, and then 1,000 women and 90 with full-term pregnancies. So we also took advantage to ask some questions on the acceptability of novel treatment if a novel therapy were to be developed, and what would be the chance of them using this novel therapy? What's the kind of characteristic these women would want to see in the therapy? And the first one was if you have an antibiotic clinically proven lactobacilli alternative, would they have a chance of using it? Quite a number of women said yes, they would have an alternative to an antibiotic to use for treatment of vaginal infection if they had. Of course, efficacious in terms of treatment was an important consideration for them to use this element. And then we also asked them about what kind of characteristics they'll be finding very important, and they reported that they would find it easy if a product is comfortable, easy to use, was made of natural products, and improve their health. And of course, a product that did not cause vaginal dryness and had partner approval. Then they also did not want a compound that caused them excessive messiness or leakage. In terms of the contraceptive use among this cohort, as you can see, many of the patients had mixed contraceptive methods, ranging from some who are not using any at all to those who are using pills, combined oral contraceptive, and injections, and so forth. We did bacterial vaginosis diagnosis using the Nugent score and the AMSEL criteria. And from this cohort, about 23% of women had a score of 7+. So they had positive bacterial vaginosis. And this was comparable to what we also did using the AMSEL criteria. About 22% had bacterial vaginosis on this particular score. So the kind works. We're now extracting DNA in preparation for sequencing that's going to start in the next 2 weeks or so. And the DNA we're extracting from the swabs that we stored at -80. And then the sequencing, we are doing it as per the Vaginal Microbiome Research Consortium protocol, so that we can have results that are comparable across, continents. And the future directions is to do, once we're done with the sequencing, we also do the metabolomics, and we intend to engage the metabolome to help us do the metabolomic profiles. Once that is done, we should be able to point out what are the most promising candidate metabolomes that can be used to fashion a mini panel that can be used to develop a point of care diagnostic or screening tool. And hopefully through that understanding, we can be able to get to know exactly what happens in pregnancy and the changes that can be used to design a library-therapeutic product. And of course, generally also understanding the mechanism of adverse pregnancy outcomes. So thank you very much for listening to me. These are some of our team members in our lab here in Nairobi. And this is the end of my presentation. Thank you.
Oh, fantastic. Thank you so much, Prof. Obingo. I was wondering, are there any questions? I don't see anything in the chat yet, but are there any questions on the talk that was just given? Thanks very much for keeping to time as well. It's fantastic. Okay, I have a question. So I was wondering, just generally looking at the landscape of microbiome research in Kenya, I think this is the first we've heard, I guess, in terms of the vaginal microbiome, which I think we'll have a few of today. And what do you think the prospects are like? So I look at you looking at, I saw you talked about the education levels, the age, and all of that. Do you, how much of an impact does that have in terms of the women you are screening? And what does a level 5 hospital mean? I didn't quite understand what that meant in terms of relative to what we see internationally. What does a level 5 hospital mean? Maybe I'm just a bit naive on that.
Sure. All right. Thank you very much. Yeah, so let me start with the level 5 hospital. So level 5 hospital is what we call a county level or regional referral hospital, where patients will be seen from a lower level or a sub-regional level hospital before they get to a level 6. Level 6 means is a national referral hospital. So if you have a region that gets patient, that hospital will be known as a level 5 hospital. So by and large, then you receive patients of different ethnicities, from different tribes. And so that gives you a wider range of cohort from the patient level. In terms of what we are likely to find, so very little is known about the microbiome, especially in an African context distribution in pregnancy. And yet for you to have healthy pregnancy and good outcomes in pregnancy, you need a healthy microenvironment, suggesting that if you have an unhealthy area, then you're likely to experience some of these adverse pregnancy outcomes. Now, it's been shown through other studies that about, between 40 to 73% of women would actually have infections, but they do not know they actually have these infections. So they are said to be asymptomatic, they have asymptomatic vaginal infections, which ordinarily do not need any treatment. But it would be very interesting to know how these infections that are symptomatic have impact on pregnancy, and also how they have impact on the general microbiome. So we hope the kind of data that we get can inform, what do we need to do for the women, first of all, those who have symptomatic infections, and number two, if we have a critical mass that actually needs some level of attention in terms of getting them to be screened early or getting them to be treated in good time as well.
Thank you so much. I have a few more questions, but I'll let you attend to some of the questions in the chat. I'll read out just one, the first one, and perhaps we'll move on and probably just have you answer the rest by just typing into the chat. But the first question here for you is, excellent talk. How do you plan to translate the results into clinical practice?
Yes, thank you very much, Sumaya. Yeah, so as I mentioned in my work, that first of all, we want to combine all these data sets, from the sequencing data to the metabolomic data and clinical data, and have an AI predictive model, create an AI predictive model that can be able to tell which kind of characteristics of women who are likely to be at risk of certain adverse pregnancy outcomes. So, for example, based on the data sets that we get, we probably would get a certain geography or certain age or certain characteristic of these women with a particular entity with dysbiosis that speaks to adverse pregnancy outcomes. So we can translate that data directly to prediction of women who are likely to be having adverse pregnancy outcomes. The second one is from the metabolomic data, we intend to get promising metabolites. And so we don't know, this is really exploratory, and we're going to be doing untargeted metabolomics. And depending on what we get, it's possible that we get repetitive metabolomic profiles in patients with preterm birth or patients with stillbirth. And so this data will be useful in terms of developing some early screening or diagnostic tools to tell which are these patients who are likely to have this kind of outcomes, so that the suitable intervention measures can be taken in good time. And then, as I mentioned, just by understanding the mechanism of pregnancy outcomes in line with deranged metabolomic profiles or dysbiosis, gives us a more understanding on this biology that can help us process new therapeutic targets for such kind of patients.
Thank you so much, Prof. Bimbo. There are so many questions in the chat. Please keep them coming. Prof. Bimbo will go ahead. Hopefully, you're able to just type in responses to the questions in the chat. Sure. In the interest of time, I'm looking forward to hearing more about your project as it advances and it progresses. We hope you'll come back and give us an update at some point again in the future.
Sure, yes, I'm happy to come back again. Thank you very much for the opportunity.
Thank you. We're going to move on to some recorded presentations now. So we've done the presentations as recorded, so we can be able to keep to time. All the speakers are present, and they'll be available for a live Q&A session after this series of talks are played. The first presentation is going to be from Dr. Philip Ashton, and it's titled The Interplay Between the Gut Microbiome and Typhoid Fever, Insights from Endemic Countries and a Controlled Human Infection Model. The second one you'll see back-to-back with is Chef Omaga, Comorbidity Profiles Among Sputum Positive Tuberculosis Patients in Cameroon. The third one is going to be from Cabo Ronald Wale, Isogenic PDHR bacterial pathogens cause a lower degree of dysbiosis in the gut microbiome. And the last one for this session will be the role of intestinal microbiota in IL-22 in colorectal cancer, prospects for microbiological and molecular analysis. So we're going to, like I said, we're going to play them back to back, and please feel free to enter your questions in the Q&A in the meantime. And all the presenters, please get ready to just put on your cameras if possible and be ready to answer questions afterwards. Thank you very much. Tina, please go ahead.
Hi everyone, my name is Philip Ashton. I'm a postdoc here at the Malawi Liverpool Wellcome Program in Blantyre, Malawi. And I'm going to talk to you today about the interplay between the gut microbiome and typhoid fever. So this work has been recently uploaded to bioRxiv, to medRxiv, which you can find by Googling these words or searching with this QR code. So typhoid fever is a serious bacterial bloodstream infection. It's caused by Salmonella typhi. It's difficult to diagnose. The mortality is high, it's around 1%, and there's big problems with AMR. It spreads via the fecal-oral route, which is why we think that the gut microbiome might be playing a role in susceptibility. One way that we know that the gut microbiota can influence susceptibility to salmonella infections is via short-chain fatty acids. So these can directly prevent infections by acidifying the cytosol of Enterobacteriaceae like Salmonella. It can happen indirectly by feeding the short-chain fatty acids, feed the gut cells and keep the gut in an anaerobic state, preventing facultative anaerobes like Salmonella from growing. And this is actually supported in human. In addition to that mouse model work, these findings have been supported in human cohorts as well. So our study design is a multi-site study. We've got 3 endemic country cohorts in Malawi, Nepal, and Bangladesh, and then a controlled human infection model in the UK. So this is where volunteers are deliberately exposed to the pathogen and then closely monitored for signs of infection. And about half the participants develop disease and half don't. So we can compare between the people who do and the people who don't develop disease in order to see what parts of the microbiota might be associated with that response. And the great thing about these is we can take the samples before the person is exposed to the pathogen, so we don't have to worry about the effect of the infection on the microbiota like we do with case control studies. So again, if you want more details on the methods, then you can see the preprint, but very briefly, we did bead beating, DNA extraction, Illumina shotgun metagenomics, taxonomic analysis with Metaplan-4, functional analysis with GutSmash and BigMap, and the stats were done with Maslin-2. So firstly, the taxonomic analysis, there were 4 species that were associated with health in both Bangladesh and Malawi. So these were a Clostridium species, Prevotella cocciprate A, a MAG, the meta-genome assembled genome in the Metaplan database, and Haemophilus parainfluenzae. So these 4 species were associated with health in Malawi and Bangladesh, but none of them were present in the majority of participants in the CHIM. So this really limits our ability to identify whether these species are associated with health in the CHIM. And this is due to differences in the gut microbiota population between the UK and our endemic country settings. So now then the functional analysis. So there was these 4 metabolic gene clusters that are all linked with short-chain fatty acid metabolism or associated with health in Malawi. and this bottom metabolic gene cluster was associated with health in Bangladesh. And then in the controlled human infection model, there was also higher amount of this metabolic gene cluster associated with short-chain fatty acids in people who were not susceptible to disease than who were susceptible to the disease. So in total, short-chain fatty acid metabolism was associated with health in Bangladesh, Malawi, and the Chin. So there's common taxonomic signals that are associated with health in endemic countries. And these included Prevotella clade A and Haemophilus parainfluenzae, which are actually both associated with inflammation in high-income countries. These are pathobionts associated with things like rheumatoid arthritis. And so where it might be a pathobiont in a high-income setting, in a high infectious disease burden setting, like our endemic country cohorts, these things may perhaps be beneficial. They could prime the immune system to respond more rapidly to infection, for example. So something else to explore there in the future. However, these taxonomic signals were not replicated in the CHIM due to the different populations in different parts of the world, different gut microbiota populations. So the functional analysis, however, did identify that short-chain fatty acid metabolism was associated with health in Bangladesh, Malawi, and the CHIM. So just very briefly, Nepal had some, it seemed slightly irregular data and didn't show any consistent signals. So in conclusion, short-chain fatty acids seem to be important for preventing infection in our cohorts. CHIMs in endemic countries would be more powerful. They'd enable us to investigate taxonomic signals as well. And functional analysis is important for combined dissimilar cohorts. So I'd just like to thank all of these people for their contributions to this large project, and I'll thank you for your attention.
I am Chefo Maga, a PhD student at the University of Buea, Cameroon, and a research fellow within the Tuberculosis Genetics in Africa project. On behalf of our research group, I would be presenting our work on comorbidity profiles among sputum-positive tuberculosis patients in Cameroon. Tuberculosis is caused by the Bacillus Mycobacterium tuberculosis, and it's usually transmitted when an individual with active pulmonary tuberculosis coughs, sneezes, or speaks, releasing aerosolized droplets containing the infectious tubercle bacilli that are inhaled by an uninfected person. This infection could either be controlled by the immune response to stay as a latent infection or progress to active tuberculosis disease. Non-communicable diseases that last for a long duration are a result of a combination of genetic, physiological, environmental, and behavioral factors. Comorbid non-communicable diseases like diabetes, hypertension, cardiovascular diseases, and kidney diseases could have implications for tuberculosis treatment management and increase the disease burden among active tuberculosis patients. Non-communicable diseases and tuberculosis share common risk factors like smoking, alcohol abuse, physical inactivity, and inadequate vegetable and fruit consumption. The main objective of this study was to profile comorbidities among sputum-positive tuberculosis patients in the southwest and littoral regions of Cameroon, relevant for improving disease management and public health interventions. This was a cross-sectional study carried out among 549 newly diagnosed sputum-positive TB cases greater than or equal to 15 years of age. Our study findings revealed 56 sputum-positive TB cases with at least one non-communicable disease, hence a prevalence of 10.2%. Diabetes was the most frequently recorded non-communicable disease among study participants, followed by kidney disease, hypertension, and cardiovascular disease. 3 out of the study participants had all 4 non-communicable diseases investigated for. From our multivariate logistic regression analysis, we could find that increasing age was associated with having at least one comorbidity. Educational level was also seen to show some significance. However, alcohol intake and body mass index were not associated with having at least one comorbidity. Sputum positive TB patients in this study greater than or equal to 45 years of age were more likely at risk of having at least one comorbidity as compared to those who were between 15 to 24 years of age. Other studies have reported older age as a risk factor for tuberculosis and diabetes comorbidity. Increasing age has also been associated with hypertension in tuberculosis patients. Contrary to the findings of this study, overweight and obesity have been reported to be significantly associated with diabetes in pulmonary tuberculosis patients. Body mass index has also been associated with hypertension in tuberculosis patients. Also, contrary to the findings of this study, HIV status and alcohol intake have been reported by other authors as factors associated with comorbid hypertension in tuberculosis and diabetes patients. In conclusion, concurrent diagnosis of non-communicable diseases among TB patients is highly recommended, as this will shed more light on these overlapping disease conditions and might support disease management and control. The references used are here cited. We would like to acknowledge our partner institutions, the Amma Hansen Research Institute, Addis Ababa, our institution, the University of Buea, Cameroon, the Tuberculosis Genetics Project, as well as our sponsors and funders, the Wellcome Trust, the Science for Africa Foundation, and H3Africa. Thank you.
Good day, everyone. My name is Kabowale from the University of Botswana. I'm here just to give you a short synopsis of my research titled Metabolism Meets Virulence. Isogenic PDHR bacterial pathogens causes a lower degree of dysbiosis in the gut microbiome. And just to give a short background on enteric infections, enteric infections caused by enteric pathogens have a high mortality and morbidity. And in 2019, there were 6.6 billion incident cases and 98.8 million prevalent cases of enteric infections, resulting in about 1.75 million deaths. And the greatest burden is excessively high in the global south, as you can see from the image here. This mostly includes Africa and other Asian countries. The etiological agents of enteric diseases include mostly bacteria and viruses, but also parasites do cause enteric infections. And in this study, we used Citrobacter rodentium, which is a mouse pathogen, to actually model enteric infections, and particularly looking at enterohemorrhagic E. coli or EHEC infections. We have previously shown that the pyruvate dehydrogenase transcriptional regulator, PDHR, which plays a role in carbon metabolism in enteric pathogens, also controls virulence in vitro in these pathogens. So to test this narrative in vivo, we used about C mice, and what we did, we deleted the PDHR in C. rodentium pathogen and infected mice with the wild type and the mutant strain which lacked the PDHR gene, and collected fecal samples from day 1 to day 21, of which were subjected to microbiome analysis. So in essence, we just did fecal microbiome analysis from these two groups of mice which were infected with the wild type and the mutant strain. And what we find out was that there was a significant reduction in bacteroids, which was observed in the two groups, and also the wild-type mice group had significant increase in the phylum Proteobacteria and Chlorophyxa at the peak of infection. And an increase in Proteobacteria meant an increase in Enterobacteriaceae, which may be associated with compromised colon integrity, which led to colitis and mostly in the wild-type mice group. And at genus level, it can be pointed out that there was an upsurge of Citrobacter and Lactosporoceaceae in the wild-type group as compared to the PDHR group. Lactosporoceaceae species degrade mucin glycoproteins and enhance pathogen translocation, and hence been linked to inflammatory bowel diseases and Crohn's diseases. So this data basically just implies that PDHR might play a significant role in enteric pathogen expansion during early aids of infection and at the late infection stages. From the 16 years microbiome sequence data, we further performed functional predictions for enriched pathways in the wild type versus the PDHR mutant group. And from this data, we showed that microbiota pathways involved in generating or degrading amino acids, carbohydrates, and energy metabolism were significantly enriched in the wild-type mice group at 9 days post-infection. Pathways involved in biosynthesis of enterobactin and enterobacterial common antigen were also increased in the wild-type group at 21 days post-infection. A few pathways were enriched in the PDHR mice compared to the wild-type group. This included energy metabolism and amino acid metabolism, and increased microbiota functional pathways in the wild-type mice group at the peak of infection suggested a response by gut microbiota to wild-type C. rodentium virulence factors. And the take-home message would thus be disturbing a pathogen's metabolism landscape that permits successful colonization by deleting these transcription factors. As in our case, we deleted PDHR severely attenuates C. rodentium virulence in vivo. An infection with this PDHR mutant strain showed a lower degree of dysbiosis in the microbial community with no significance at the peak of infection and at the end of infection. And thus, PDHR could be used as a potential target for the development of a safe therapeutic for controlling enteric pathogens. And thank you for your time. Thank you for listening.
Good morning. I am Wafa Baba and I am going to share our research on the role of intestinal microbiota and interleukin 22 in colorectal cancer. Colorectal cancer is one of the most common cancers worldwide. In 2022, 1.9 million new cases were diagnosed with over 900,000 deaths. In Tunisia, colorectal cancer ranks third in terms of incidence after lung and breast cancer with 1.8 thousand new cases and 953 deaths. Colorectal cancer is a complex multifactorial disease influenced by genetic and environmental factors. Recent research suggested that intestinal microbiota may play a significant role in carcinogenesis development. Gut microbiota is a complex ecosystem of microorganisms that live symbiotically within human digestive tracts, contributing to nutrition, metabolism, and barrier functions. So, development and progression of CRC are influenced by physiologic state and composition of intestinal microbiota, which affects the immune response and involved in carcinogenesis. In this context, our study focused to identify and analyze specific bacteria species in gut microbiota of patients, and to explore potential interactions between bacteria and interleukin 22. To achieve this, we performed microbiological and molecular tests on tissue samples and swabs from 12 colorectal cancer patients and one healthy control. We used a range of microbiological and molecular tools such as bacterial culture and central sequencing to identify the present species. The qPCR was used to investigate interleukin-22 expression gene levels. Bacterial strains from Lactobacillus and Enterobacteriaceae strains was identified. Molecular identification confirms the presence of 2 strains of Enterobacteriaceae strains and 5 strains of Lactobacillus genes in CRC patients. After PCR amplification, Fusobacterium nucleatum was found in most tissue from CRC patients, 9 malignant tissue and 7 healthy tissue. On the other hand, interleukin-22 gene level was highly expressed in healthy tissue compared to tumor tissue. It was also increased in healthy tissue compared to tumor tissue with Fusobacterium infection. We found also that interleukin-22 gene expression was increased in Fusobacterium infected tumor tissue compared to uninfected tumor tissue in CRC patients. In conclusion, gut microbiota is a promising area of research against CRC. In this study, we identify some lactobacillus strains such as Lactobacillus paracasei and intestinal mucosa of CRC patients. Several studies suggest gut dysbiosis in CRC patients characterized by an increase of pathogenic bacteria. Our study identified Klebsiella pasteurizii strain and confirmed the presence of Fusobacterium nucleatum. Interestingly, interleukin-22 gene expression was increased in Fusobacterium nucleatum infection tumor tissue compared to uninfected tumor tissue in CRC patients. This result suggested that gut microbiota modulation could be a promising therapy against colorectal cancer. By modulating gut microbiota composition, we could potentially open new therapy ways. This approach might lead to personalized prevention and treatment strategies for CRC in future. Finally, I would like to thank our collaborators and partners, especially Dr. Wael Ferjawi and Dr. Mohammed Haji and the team members at the Laboratory of Microorganisms and Active Biomolecules. Thank you for your attention.
Thank you very much to all the speakers. We've really kept to time. I do see a few questions in the chat. Let me see. Are there any questions for? There are a few questions for Dr. Ashton, but he's joining shortly. But in the meantime, I would like to ask, Shefor, are you on this call? I'd like to just ask Shefor, in your project, in your study on tuberculosis, was it something you expected generally in Cameroon? Because we know that there are certain areas that we kind of know tuberculosis to be, this is a more general question, to be quite endemic in. When you started out on your research or got on this project, was there any surprises? Was there any surprise for you as you went through the process of your research with respect to numbers you found and what you found?
Okay, thank you very much for this question. Actually, tuberculosis is a major concern in Cameroon, and these findings were not very strange, because we see that the prevalence of tuberculosis reported in this study is is in line with other studies that have been carried out in the country. What was striking was the prevalence of diabetes, these comorbidities in these tuberculosis patients. I don't know if I answered your question.
You did. I guess I'll ask a follow-up question. Why was that striking? Why do you think that was striking? Not to put you on the spot, but why do you think that was striking?
Yeah, because diabetes is not concurrently diagnosed in patients who are known to have tuberculosis. But if these diseases, these non-communicable diseases are concurrently diagnosed, we will be able to clearly say the prevalence rates and how they affect these patients. And that was why in our conclusion, we were suggesting that concurrent diagnosis of non-communicable diseases should be incorporated in the health system for these patients who have been diagnosed with tuberculosis, since it's an infection that can be cleared only with the long-term treatment of 4 to 6 months. And it comes most times with a lot of side effects, abandoned treatments, and multi-drug resistance.
Thank you very much, Chef, for brilliant work. Dr. Philip Ashton, there are a few questions for you. The first one is from Boniface Kichuki. He says, great talk. Were there short chain fatty acid profiles associated with health specific to particular species or strains, e.g. Prevotella copri?
Hi, thanks for the question. So yeah, there was we did the functional analysis on 2 levels. Firstly, looking at specific metabolic gene clusters that kind of belong to a single species. And as you would kind of expect, those findings mirrored the taxonomic findings because you're just looking at the same data in 2 different ways. So a lot of those species were from Prevotella copri, Haemophilus parainfluenzae, and the other taxonomic, the other species that we identified. And then we also looked, we combined, based on the metabolic gene cluster type, we combined all of the reads that were assigned to that metabolic gene cluster type, and did the statistical analysis on that level as well. So on both occasions, on both analysis types, short chain fatty acids flagged up as one of the things that was associated with health. Short chain fatty acid metabolism gene clusters that were associated with health. So yeah, I think that's fairly robust. I think that's a fairly robust finding.
All right, thank you so much, Dr. Ashton. Just to say thank you for the questions. There are a few more questions in the chat. Please feel free to continue asking your questions, and presenters, please try and respond to the questions in the chat. Sheffer, there's one for you in the chat as well, and I think there's a few more for you, Dr. Ashton, in the chat. So thank you so much, everyone. In the interest of time, we're moving on to the next session, and I'll hand over to Professor Lamek Mapaga.
Hi everyone. I hope you can all hear me. Right.
Yes, we can. Thank you. Please go ahead. But we don't see your did you have your video on, your camera on, because we can't see the black screen, but that's okay too.
Okay. No, it's fine. I hope now I can be seen. Right. Can you see now?
Not really, but that's okay. Please go ahead.
Okay, I'll just go ahead till come up maybe. Right. So okay, yeah. So thank you very much. So thank you very much, Voke, and for everyone who has joined the sessions. So this takes us to section 2 of the event, where the next speaker is Dr. Harris Onywera. Dr. Harris Onywera is a bioinformatician, data scientist at the Africa CDC. His research interests include molecular biology and epidemiology of human infectious diseases, pathogen genomics, microbial drug resistance, microbiome in health and disease. And he has previously worked at various institutions, University of Cape Town, South Africa, Center for Genomic Regulation, CRG in Spain, and US Army Medical Research Institute, Walter Reed Project in Kenya, and also plus at the Kemri CDC and also in Kenya, and also worked at Kisumu. So he earned his PhD in medical virology from the University of Cape Town, holds MSc in bioinformatics and computational molecular biology, and BSc in biochemistry and molecular biology. He also has degrees from, of course, BSc chemistry from first class from Rhodes University. So the talk that he's going to take us through today, his talk is going to be on cervical microbiota and HPV infection in productive age African women, insights from retrospective cross-sectional study. Thank you. Dr. Harris, please, you can start sharing your screen.
Good afternoon, everyone. Please confirm that you can see my screen and hear me.
Yes, we can see your screen and hear you.
Okay, thank you so much, Dr. Lamek, and organizers of this virtual conference. It is a privilege to be part of this audience and invited speakers. So my talk today will be on cervical microbiome and association with HPV. So this is a retrospective cross-sectional study based on previous work that I did at the University of Cape Town. Before I proceed, I'd like to acknowledge everyone that was very instrumental in this project, from my supervisors, collaborators, study participants, and the funding agencies. By way of introduction, we all know that a balanced microbiome is essential for sustaining homeostasis, and disruption of this microbiome actually is linked to development of diseases. Microbiome has a critical role in One Health, and I'll just give a highlight on what this one entails. Actually, microbiome has been shown to regulate the immune system, and it can also be used to prevent human infections. Also, microbiome has been found to be a critical reservoir for antimicrobial resistance genes. Microbiome is also impacted by the quality of water, pollutants, and agricultural practices. And if there is perturbation or disruption of the environmental factors, then they are likely to perturb the microbiome, and this can end up creating a feedback loop that will eventually impact the human and ecosystem health. Another area where microbiome is important is on food safety and security. It's known that microbiome affect both the digestion and absorption of nutrients, linking these to food security. And lastly, microbiome has also been shown to be critical, and it can be used to serve as an early warning indicator for emergence of zoonotic diseases such as Ebola and so on. Of course, there are other applications of microbiome in human health. So in the context of this talk, it's been shown the exploration of the bacteria in the cervical and vaginal environment began in mid-1800, and to date, over 250 bacteria have been characterized using culture-independent technologies, specifically next-generation sequencing technologies. And looking at the human vagina and cervix, it's been found that it is composed of a stratified squamous epithelium that is lined with mucosa, and this mucosa is rich in glycogen layer. This glycogen layer is the one that supports the existence of the microbial communities in the cervicovaginal tract. This microbiome used a number of mechanisms, actually antagonistic mechanisms, to foil or to prevent other bacteria, other pathogenic and opportunistic bacteria from colonizing the cervicovaginal tract. And some of these mechanisms include production of lactic acid, which is associated with low vaginal pH, and low vaginal pH creates an environment that is not conducive for both pathogenic and opportunistic bacteria. There's a big study that was done in North America, and the finding from this study was that the vaginal microbiota of reproductive women is influenced by ethnicity or race. So in this study, they included a total of 396 women from Asian, Caucasian, Black, and Hispanic communities. When the microbiome analysis was performed, they found that these microbial communities could be categorized or classified into 5 community state types. The first community state type was denoted as the one that is colonized by Lactobacillus crispatus, the one in green. We have Lactobacillus gasseri, and so on. And we have a community that is not dominated by any of the keystone Lactobacillus species, and this community is referred to as a diverse community. From this study, they found that a majority of the Black women actually had a cervicovaginal microbiota that was not colonized by any particular Lactobacillus. So from this, you can see these women were colonized by a diverse group of bacteria that have been associated with bacterial vaginosis, which is the most common form of vaginal disorder among reproductive age women. The other finding of this study was that they also looked at the vaginal pH in these categories of women: Asian, White, Black, and Hispanic, and they found that Black and Hispanic women had a higher vaginal pH compared to both Asian and White women. And this study actually has been consistent with subsequent findings that have found that at least 1/3 to 2/3 of African women have an lactobacillus-dominated environment, and most of the Black women, they have a less acidic vaginal environment that is consistent to lack of the keystone lactobacillus, that is Crispatus gasseri, Inus, and Jensenii. The other thing I'd like to point out is that increased vaginal pH has been associated with incidence of human papillomavirus, whose persistence has been associated with cervical cancer. So what was the motivation of the present study? Studies have shown that South Africa has a high burden of HPV ranging from 16 to 75%. And some of these HPV types have been associated with cervical cancer, which is among the highest cancers in South Africa. And the mechanism or the reason why South African women have a high burden of HPV and cervical cancer is not fully understood. But by the time we were doing this study, we had postulated that there could be biotic factors, that is, the bacteria in the cervix and vagina were playing a critical role in this. Some of the available studies on cervicovaginal microbiota and HPV, by the time we were doing this study, were controversial and inconclusive. There were some studies that found no association at all between bacterial vaginosis and HPV infection. Some found a positive association between BV and HPV in terms of the prevalence of HPV incidence, and at the same time, the presence of BV was associated with the delayed clearance of HPV. Remember, HPV persistent has been associated with cervical cancer. Some studies have found weak positive association between some of the bacteria in the cervicovaginal environment and HPV infection. And by the time we're doing this study, there was limited information about the cervicovaginal microbiota of South African women and some of the factors that were impacting these bacteria. So the aim of this study was to profile the cervicovaginal microbiota of reproductive age South African women and to find out whether there were any association between these microbiota and the metadata, particularly HPV infections. This was a retrospective cross-sectional study that was nested within the HPV couples cohort study. So in this study, the aim of this HPV couple cohort study was to look at the concordance and discordance of HPV among Black South African couple. So for this study, we used part of the stored cervical specimen. Using these samples, we extracted the DNA. These samples were, we prepared libraries, which were then subjected to Illumina MySeq sequencing. From the data that we got from this sequencing device, these were subjected to bioinformatics analysis using bioinformatics and statistical tools, and the pipeline that we used to assess the microbial community was CHIME. There were other analyses that we used to assess the association between the cervicovaginal microbiota and the metadata. So this is the first result. So you can see from this table, we had a total of 87 female participants, and these were women of reproductive age between 18 to 44 years of age. A total of 43% of these women had were positive for HPV, and 34.5 had at least a high-risk HPV infection. We also have information about the cervical cytology of these women. A majority of them had a normal cervical cytology. We also have information on bacterial vaginosis, and about 44% of these women were positive for BV. So for this study, once we got the genomic sequences of the bacteria, we did a hierarchical clustering, and we found that these women, the microbial communities of these women could be stratified into 8 community state types from 1 to 8. So from this heat map, you can see on the vertical axis, these are the bacteria, the topmost bacteria that we detected, and each column is a particular woman. You can see information on the variables. So we have information about women that were positive for BV and those that were not positive for BV. So anything in black is an indication of positivity. Anything in a shade of gray is negative. So for HPV, you can see anything in black again, it's positive for HPV. Then we have a key map. We have this key. So the more abundant a bacteria is, the hotter the scale. So anything in towards blue or white means that the relative abundance is lower. So from this study, you can see that the microbial communities were clustered into 8 community state types. Community state types 1, 2, and 3. These were dominated by lactobacillus species, and we have the rest that were not dominated by lactobacillus. So the key message from this study was that a majority of the Black South African women that we studied had a cervical environment that was not dominated by any of the common lactobacillus species. So how does our study compare to the previous study that was done on the North American cohort? So in our study, we found that in addition to some of the lactobacillus species that were detected, we found other communities that were dominated by other bacteria, for example, streptococcus, chlamydia, and so on. Then we found that in our study, lactobacillus, which is not the these are bacteria that has a capability of. In other studies, it has been found that it is associated with sexually transmitted infections compared to other lactobacillus species, which are known to be more protective. So in our study, we found that among the lactobacillus species, Lactobacillus inus was the most common. And in Cape Town, where our study was done, the prevalence of this lactobacillus species was 83%. compared to a study that was done in KwaZulu-Natal, which showed that the lactobacillus illness, the prevalence was 77%. In terms of the diverse communities, we found that there was no difference between the study that was done in Cape Town and in KwaZulu-Natal. The next thing that we assessed, we wanted to find out whether we could classify or categorize these microbial communities according to the community state types. We found that there was a clear separation in both alpha diversity and beta diversity in terms of the community state types. The community state type that is dominated by lactobacillus, that is community state, particularly this one, CST1, we have CST2, CST3. On average, these cervicovaginal communities, they had lower bacterial diversities compared to the diverse community, that is the one in red. So again, you can see that the communities that were dominated by lactobacillus species, they clustered together, whereas the ones that were dominated by the diverse communities, actually they spread out through the three-dimensional space. We also did the same analysis to assess whether bacterial vaginosis could be used to cluster these bacterial communities, and we found that there was a clear separation between women that were positive for BV versus the ones that were negative for BV. In terms of HPV, there was no segregation. So by this, I mean that we could not classify, or we could not find any differences in bacterial diversity between women that were positive for high-risk HPV versus without high-risk HPV. The next question that we asked ourselves was whether there were specific bacteria whose abundances were differentially abundant in women with versus without HPV. So from these results that was done using LEFC or linear discriminant analysis, you can see that there were some bacteria that were more, whose abundances were more pronounced in women without HPV, and others were more pronounced in women with HPV. So then bacteria that we found that could be used as strong biomarkers for HPV. These were Schnitzia, Atopobium, and Gardnerella species. So what we found is that I'm only going to talk about this Schnitzia species. This Schnitzia species has been found to be a common biomarker, especially for women in South Africa, Nigeria, Italy, Korean, and Chinese. And this bacteria has been associated with increased vaginal pH, cervical intraepithelial lesion. At the same time, it has also been associated with genital inflammation and cervical neoplasia. And lastly, there's some information that some of the genes encoding for the proteins in this particular bacteria, that is Schnitzia, have been associated with cytotoxicity and adherence to cervical cells. So the conclusion of this study was a majority of the women had a cervicovaginal microbiota that is not dominated by lactobacillus. And we think that more in-depth studies are required to find out the biological mechanism, maybe explaining the interaction between this Schnitzia and HPV infections. Thank you.
Thank you very much, Dr. Harris, for a very clear and wonderful presentation. It was very audible, and we could clearly follow what your research was all about. Right. So because of time, I think you can all share your questions on the chat, and then Dr. Harris will be able to address all the questions that have been raised on the chat. So there's one question, Harris, says from Nabil Amor, dear Harris Onywera, are the V3, V4 the most accurate 16S rRNA hypervariable region for taxonomic identification?
Yes, there are some studies that have been carried out to assess which of the 9 hypervariable region of the 16S could be used as an optimal region to do microbial analysis. And to date, there is no region that has been satisfactorily found to be an ideal But based on the studies that have been done, they've found that V3, a combination of V3, V4 will be ideal because it's able to give, in terms of classification, you're able to go into higher depth of taxonomic classification. That is, you're able to find species that you're able to identify species and genus. But there are some regions, say for example, V3, if you use V3 for microbial analysis, you might not be able to go into the deeper taxonomic levels. You might only be able maybe to reach up to the family level. Over to you, Dr. Lamek.
Okay, thank you very much. Maybe one last one before you maybe address them from the chat, and then we move to the other oral presentations. Have you tried a few of the samples on shotgun? This is a question from Gildas.
None at the moment.
Okay. Thank you very much, Dr. Harris. So because of time, we'll have to move on to the next oral presentations, where we'll have, these are recorded presentations, so you can be able to also write your questions on the chat once we are done with the oral presentations, and then the presenters will be able to address each of your questions and concerns. So we have Eunice Njuguna, Lara Helmi, Habiba Osman, Rima Ahmed, Salma Ahmed, Habiba Aldeu, Sarah Faljah, and Sarah Gaida. Over to you, Tino.
My name is Eunice Njuguna. I am a PhD candidate working with the following institutions. And today I'll take you through part of my PhD work where I'm looking at the impact of pneumococcal conjugate vaccine, PCV10, on the pneumococcal population structure in Kilifi, Kenya. And this is a whole genome sequencing analysis. So, invasive pneumococcal disease, IPD, has a high burden in sub-Saharan Africa, and surveillance in this setting is also limited. And we are available, for example, in Kenya, it's in the based in research centers. This translates to the available data and even the genomic studies that are available. So the current tool that is being used to control IPD globally is PCV, pneumococcal conjugate vaccines. And the current problem that we're having, or we are facing, a serotype replacement, whereby as we continue to use the vaccine serotype are eliminated from the population, but the non-vaccine serotypes are now causing the disease. And this has been evidenced in high-income countries where they have a routine pneumococcal surveillance. In Kenya, the first PCV vaccine was introduced, PCV10, with the following serotype listed here. So these were my vaccine serotype, and this led to a 92% decline of vaccine-related IPD. However, it was predicted that 52% of there will be a 52% increase of non-vaccine related IPD 10 years after introduction of this vaccine. So there is need to understand the evolution and what is happening to and leading to evolution and non-vaccine serotype causing the disease. So the baseline is to understand the genomic population structure in this setting. So the study aims to look at the impact of pneumococcal vaccine by characterizing the genomic population structure of pneumococcus before and after vaccine introduction in Kilifi, and this was done using a data that was collected from year 2003 to 2017 cross-sectionally, and was done on children and adults of between 0 to 94 years. Disease and encouraged samples were used, which were selectively cultured for SPN using gentamicin media. SPN is Streptococcus pneumoniae, and quelling was done to serotype them, and then the single colonies were extracted and sequenced using Illumina HiSeq platform. The samples or the reads were analyzed as follows. The orange highlights are representing the respective tools that were used. So quality check was done using FastQC tool and Kraken, and then lineage were classified using PopPank, and Serobar was used to serotype. AMR profiling was done on 17 different microbials using CDC pipeline, and maximum likelihood phylogenies were constructed using FastTree and visualized in MicroReact. This is the summary of the demographic. Much of the data lies on Courage and children of less than 5 years. So onto the serotype distribution, a total of 58 different serotypes were identified in this population, and divided in Courage and disease. In Courage, First of all, we have on the y-axis, we have the proportion, and on the y-axis, we have the serotype, and different colors are showing, the grays are representing vaccine serotype, and the blues are showing non-vaccine serotype. So before vaccine introduction, in courage, we had 19F as the most prevalent, and 19A post-vaccine, and in disease, we had serotype 1 as the most prevalent before and after vaccine introduction. The same case orientation as for the serotype here in the lineages. We recorded 132 different lineages, and 30 of them were novel, not recorded elsewhere globally. And then the colors and orientation are the same as the serotype, with the green, an extra color with greens, which are showing the lineages that were mixed with vaccine and non-vaccine serotype. So in disease, we had GPSC2 prevalent before and after vaccine, and in courage, we had GPSC 879 before and GPSC 22 after. Looking at the significant changes, we had 11 significant change in courage, and in disease, we had serotype 15A and serotype 1 with significant change. And in courage, we had 10 different serotypes that had significant changes, that is in GPSCs, and GPSC 22 and 2 were significant in disease. Important to note that GPSC 22 had 9 different serotypes, and out of 9, 6 of them, the ones circled here, had a significant change. This led us to look at GPSC 22. So looking at GPSC 22, it is quite divergent with some different clustering, and we can see that it's characterized with penicillin resistant in most of the isolate, and penicillin is the one used to treat IPD in Kilifi. And also we can see some recording of multi-drug resistant on some isolates. And looking at the recombination level, so this is a recombination profile. The red are showing the recombination levels, and you can see much is happening in this lineage, showing how it's evolving to characterize to be able to evolve post-vaccine. Also recorded some multi-drug resistant lineages in this lineage. And also in summary, we were able to give the impact of the vaccine the impact of this vaccine, and also recorded some novel lineages and multi-drug resistant lineages, and also serotype replacement was evidenced. I would like to acknowledge the following individuals and institutions for their
Hello, my name is Sarah Farjala from the Department of Biology at the University of Tunis Al-Manar. I'm presenting my work research entitled Assessment of the Genetic Structure of Lamlodiscus echenis in the Mediterranean Sea Indicates Parasitic Transmission between wild and cage-reared Sparaesorata populations. Aquaculture in the Mediterranean Sea has become essential in the food sector due to the decline of wild fish stocks and is recognized as the major contributor to seafood for human consumption. The gilt-head sea bream Sparaesorata is a common fish species throughout the Mediterranean Sea and the Eastern Atlantic Ocean. As a fish species of significant economic value, Turkey, Egypt, and Tunisia represented more than 60% of the total production. The diplectenid monogenean Lamlodiscus echenis is a specific and common gill parasite of wild and cage-reared gilt-head sea bream in the Mediterranean Sea. Despite the significant prevalence of the species and its widespread distribution in the Mediterranean Sea, only a few isolated molecular studies of the species have been conducted. So, in the present work, the population genetic variation of Lamlodiscus echenis isolated from wild and cage-reared Sparaesorata from several Mediterranean localities of Tunisia, Italy, and Spain is evaluated using nuclear ETS rDNA and mitochondrial cytochrome oxidase 1 markers, and its phylogenetic status is described. Specimens of Lamlodiscus echenis were collected on the gills of 160 specimens of Sparaesorata from 15 localities of Tunisia, Italy, and Spain. After DNA extraction, we proceed to amplification of molecular markers using Bantolab, and PCR products were sequenced at Microbiome Europe facilities. For phylogenetic and population genetic analysis, we used MEGA, DNASP, PopArt, and Arluca. The phylogenetic analysis using the partial fragment of the ETS rDNA was consistent with previous studies on Lamlodiscus echeneis and provided us to the molecular characterization of Lamlodiscus echeneis. The 57 theory haplotypes found in the study reflected a high genetic diversity of the species within the survived area. The haplotypes shared between wild and farmed hosts provided an insight into the population genetic variation of Lamlodiscus echeneis, confirming the potential for cross-infection between wild and farmed hosts. The star-like haplotypes network suggested a recent population expansion of Lamlodiscus echeneis. This is in line with the observed high haplotype diversity and low nucleotide diversity. This result was confirmed also by the AMOVA, where more than 65% of the total variation occurred within populations. Based on COE sequences, phylogenetic analysis grouping all samples into the main clade corresponding to Lamlodiscus echeneis from several localities revealed no clades that could be associated with any geographic pattern. In conclusion, the study provides insight into the population genetic variation of Lamlodiscus echeneis, showing an absence of genetic selection and a high level of gene flow through Tunisian, Italian, and Spanish localities. The observed patterns are most likely caused by recent demographic expansion. On the other hand, the haplotypes shared between wild and farmed hosts confirmed the potential cross-infection, providing molecular evidence for pathogen transfer. Thank you for your attention.
Good evening, everyone. I'm Lara Helmi, a student at the Faculty of Medicine at Galala University, presenting together with my fellow students, Habiba Osman, Reem Ahmed, Salma Ahmed, and Habib Al-Digwi, under the supervision of Professor Dr. Reem Salam, Faculty of Medicine, Galala University, and Lecturer Dr. Lubna Nabil, Faculty of Medicine, Ain Shams University. On behalf of the group, I'm honored to present to you our work titled Fermented Food Consumption and Gut Microbiota in Egypt, Current Challenges and Future Directions. These are the aspects we're covering in our presentation today. Fermented foods are everywhere, and they've been there for a very long time. They're considered to be among the first foods consumed by humans. Over thousands of years, humans have been using the process of fermentation throughout civilizations to preserve and produce a variety of culturally significant foods. Some of the earliest documentations of such exist in ancient Egypt, where fermented foods were an essential part of daily meals. To this day, fermented foods still constitute a part of the diet of most people in Egypt.
Of the most common examples of these foods are fermented dairy products, roughly all of which are based on the most basic fermented product, sour milk, or laban raib as we call it in Egypt. Sour milk is prepared from fresh milk by leaving it in a warm place until the cream separates from the liquid fraction of the milk. The cream layer is used as butter, and the remaining liquid is the sour milk. We also have laban zir, which is coagulated sour milk that has a thick, creamy texture and is very acidic. The most consumed fermented dairy product, however, is mish cheese, which has a strong flavor similar to that of Roquefort cheese, with 65% of its composition being moisture and about 14% salt. Then we have kirsch saidi made up of wheat and laban zir structured in small, rough, rounded pieces. Upon adding water, they turn white and break up into a delicious, creamy paste. On the other hand, fisis and muluha are Egypt's most popular fermented salted fish, which are consumed annually in ancient Egyptian holidays.
Dairy products are an ideal example on how a certain fermented product is converted to another by adjusting the earthenware pot's temperature and duration. For demonstration, under cold conditions, laban raib is extracted from milk, then poured over a special cheese mat called hasira, and left to drain for several hours, giving us Arish cheese. Further fermentation of this cheese could be done by providing a pickling medium consisting of buttermilk, laban raib, whey, and murtsa, all placed in a balasa, which is a special type of earthenware pot. covered by mud or ensuring anaerobic conditions, and left for not less than 1 year, leaving us with mist cheese. The pickling medium plays a crucial part in mist cheese fermentation process as it alters its chemical and microbial composition.
These alterations in fermented food not only extend their shelf life expectancy, but also provide a wide range of health benefits associated to its consumption, such as modifying microbial population, enhancement of the function of epithelium, modulation of the inflammatory response, as well as improving gut mucosal barrier function, all leading to decrease the risks of multiple serious health issues such as gastrointestinal intolerance, cancer, heart diseases, allergic reactions, lactose malabsorption, and decrease the risk of diabetes. Now, how do these fermented foods exactly influence our gut health? Whether directly, through supplying probiotic bacteria to compete with the inhabited pathogens, or indirectly through its degradation into other substances to produce these effects? That's a question that researchers have yet to answer.
Let's talk about some of the challenges we faced while researching fermented foods in Egypt. One of the most significant issues is the lack of a standard method for preparing these foods, and this inconsistency made it difficult to have a reliable research. Although Egypt has a rich history with fermented foods, we still need more research to understand and fully support their potential benefits. But overcoming these challenges will have substantial benefits on our health. So, what can be done? The first step is to compile a comprehensive list of the fermented foods currently consumed in Egypt. From there, a database can be created that details how these foods are prepared in various regions, allowing for more consistency in future research. And finally, the most crucial step is conducting molecular and cellular studies to determine how these foods affect our health. This will give us the scientific backing to promote these traditional practices.
This gets us to our conclusion that once databases on the diversity and composition of fermented foods in Egypt are compiled, research analyzing the links between human health, gut microbiota, and consumption of fermented food can be more thorough, therefore opening up opportunities to utilize such research in developing innovative approaches to prevent and manage several metabolic diseases. Thank you, and we're looking forward to your questions and your feedback.
Hello everyone, I'm Sarah Gaida, I'm PhD student. I have the honor to present to you my work which is entitled Assessment of Biochar and Compost Effects on Rhizospheric Soil, Enzymatic Activities and Metagenomic Diversity. The agricultural soil presents an important biological support and its decline poses significant challenge, which leading to degradation of quality and decrease in agricultural productivity. So it's important to maintain soil health, which can be achieved by using organic amendments such as compost and biochar. To study the effects of these amendments, we use Solanum lycopersicum as a biological indicator. This study aims to achieve the following objective: the assessment of the combined impact of biochar and compost on soil biofertility through the evaluation of enzymatic activities and metagenomic diversity in organic and conventional rhizospheric soils. To do this, we adopted this experimental design. The compost blocks contain 1% of soil weight, the biochar blocks 2% of soil weight, and mixture blocks, and then control blocks with no amendments. Then we plant tomato plants. We have determined the soil physicochemical properties, soil microbiological activities, and characterization of bacterial communities. The results of soil physicochemical properties revealed a decrease in pH for both types of soil and all treatments, and an increase in soil electrical conductivity for all treatments. This due to the mineral salt content in biochar and compost. The result of soil enzymatic activities showed that in biological soils, biochar and compost individually led to higher enzymatic activity compared to the mixed treatment BM. In conventional soils, compost generally resulted in the highest enzymatic activity, notably for dehydrogenase and urease. The results of microbial activity and functional diversity indicate that the compost have a positive effect with the biological farming systems. Metagenomic analysis revealed that the microbial composition across different taxonomic levels, from phylum to genus, remains consistent across soil treatments, with actinobacteria and proteobacteria dominating, though diversity increases at more specific levels. The principal component analysis shows that the separation along PC1 and PC2 reflects the major impact of the treatments on microbial diversity, and biological treatments are separated from conventional treatments, highlighting differences in soil microbiota. This graph indicates that Acinetobacter, Bacillus, and Aeromonas exhibit the highest LDI scores, indicating their importance in differentiating the microbial communities across treatments. Moving to the discussion for our findings, the results can be explained by the capacity of compost to enrich the soil with organic matter and nutrient, promoting plant growth, decomposition, and increased microbial activity. The biochar can improve soil porosity and nutrient retention, providing stable habitat for microorganisms, stimulates enzymatic activity and microbial diversity. Then the combination of biochar and compost creates a synergy that improves nutrient availability, enzymatic activity, and microbial diversity, making the soil more fertile and resilient. So, as a conclusion, the use of biochar and compost in different modes of farming affect the physicochemical properties, enzymatic activities, and metagenomic diversity. Further research would be necessary to determine the conditions, dosages, and context in which the use of biochar and compost may be beneficial to detrimental to biofertility. Thank you for your attention.
Thank you very much for the presentations. So that was the second part of this presentation in terms of microbiome and One Health general. So I'm looking at questions here, but I'm not seeing anybody who has posted any question. But you're still able to post any questions that you have to the various speakers who are actually going to be able to address any questions or concerns that you have, because all the speakers are actually online, and it will be imperative if you have any questions or concerns to raise with them. Maybe before I hand over to Dr. Brian, okay, there's one question here from Nabil Amor, but before I got that question, Eunice Juguna, you mentioned the issue of you found 30 novel lineages. Is it possible to speculate or what you think these lineages might be? What would be their role in terms of vaccines? What you're looking at? Eunice, are you available? As Eunice is thinking about that, there are a couple of questions by Nabil Amor. Last presentation of Sarah G, which technique did you use for the meta-barcoding approach? Sarah Gaida. And Eunice Juguna.
Everyone mute.
Eunice Juguna, yes. Maybe let's go with Eunice Juguna first.
Yeah. What's the question, please?
Oh, the question that was being asked was you found some 30 new novel lineages in your study.
Yeah.
Are you able to speculate what their role are in terms of vaccines, the study that you're looking at?
No, actually, that is something that I'm currently working on, because I had to forward the lineages to just update the lineages and to the database. But looking at patterns that I can be able to like to look to differentiate with the rest of the lineages. They are blend in and they are distributed all over. So I cannot specify anything that they have anything unique. They are just different lineages that have not been recorded elsewhere.
Okay.
Yeah.
Exciting. So maybe in the next African Microbiome 4.0, maybe we'll get to know more about those lineages and other new ones. Right. Is Sarah Gaida, are you there? Sarah Gaida? Maybe you need to unmute. Okay, maybe Sarah Gaida is going to respond. Okay, yes, she has responded. Sequencing specific genetic markers, so she has responded online. The person who had the question has been addressed in the chat. So that brings us to the end of this section, microbiome and One Health general. I'll now hand it over to Dr. Brian Carling to tell us more about what will happen during this break. Thank you very much. Over to you, Brian.
Thanks, Dr. Simon. Yeah, so we're just going to have a brief, it's going to be a combination of a bit of a break. People can stretch their legs, maybe take a comfort break, make a cup of coffee, check emails, anything like that. But we'd also like to make it an option for people to do any networking if they want to. So we're going to stay connected, and we're not going to have specific separate breakout rooms. It's all going to happen in this central room. There's going to be 2 posters which will be shared. If the poster, the people who made the posters are present and they could indicate that they are, then this is a good opportunity to ask them questions about their posters, as well as just to maybe just meet each other. So the idea would be, in the beginning, some people said we were welcome, we hear from wherever. If anyone's still on and they want to be able to say, this is where we're from, this is what we do, you're welcome to put that in the chat. And this will go on till 4 o'clock. And at that point, we'll go into our 3rd session, which will be chaired by Professor Joanne Passmore. So could we, thanks for showing the posters. Cool. So we've got about 5 minutes before the start of the next session. So anyone who took a quick break and is coming back and is not sure what's happening, we're just having a little bit of a networking session. There's a photo up that people are welcome to watch. And we'll resume again officially at 4 p.m. In the meantime, everyone's just saying hello in the chat. So it's great to see we got people from good representation from Uganda, Nigeria, Cameroon, Tunisia, so Egypt. So we're covering a lot of Africa. It's great. It's wonderful to meet you all.
Hello, everyone. I'm going to give everyone another minute to get back to their computers with their cups of tea, and please keep the chat going. There's some adverts for upcoming events in the chat as well. So please take note of these. And it's wonderful to have such a broad representation from across Africa on the call today. So it's exactly the top of the hour. My name is Dr. Joanne Passmore. I'm based at the University of Cape Town in South Africa, and it's my absolute pleasure to host this session at the third African Microbiome Day. And I just wanted to say congratulations to Voke and her dedicated team for making this happen each year, and for all of you to support the events, and hopefully it grows from strength to strength. My task as chair of this session, which is entitled Microbiome and its Application, is to introduce our first invited speaker for today. And it gives me great pleasure, obviously, to introduce Professor Heather Jaspin, who is from the University of Cape Town, from the Division of Immunology. Heather is an amazingly talented clinician scientist who has set up microbiome research capacity at the University of Cape Town, and I've been collaborating with Heather for many years. Those who know Heather well know she's a born and bred South African with honorary appointments at the University of Cape Town, in addition to holding an associate professorship at the University of Washington in Seattle and Seattle Children's Research Institute. To give Heather the maximum amount of time to give her talk on a fascinating and highly relevant topic, I'm going to hand over to Heather to present her slides.
Great, thank you so much, Joanne. Good morning, good afternoon, everybody. Thanks for the introduction, Joanne. I don't think I need to introduce myself any further. Today, we decided I would present a story about bedside to bench and back again, which I've entitled Bifidobacterium infantis supplementation to improve immunity in infants exposed to HIV. So I think this audience knows that the early life microbiota is extremely important in immune ontogeny. And there's some evidence in humans that there's an association between the gut microbiome and their response and infant responses to vaccination, particularly humoral antibody responses, and some association previously described for the relative abundance of bifidobacterium in infant guts. The early life gut microbiota is also extremely important for other things such as neurodevelopment and metabolic outcomes, at least in mice. This has been shown quite well. So part of our lab in Cape Town studies these infants that are born to mothers with HIV, who we call HIV-exposed infants. And although they're HIV uninfected themselves, well, first of all, since the advent of PMTCT or vertical transmission prevention, we've managed to prevent infections, but there's this growing population of these exposed infants who remain uninfected. And in South Africa, for example, and in Eswatini, we have almost 30% of infants to be HIV exposed uninfected. And although these are old data, the trends still persist. These infants, despite being HIV uninfected, are at higher risk for morbidities and mortalities. Usually these are due to infectious morbidity, but they also have altered neurodevelopment and growth in some instances. And we think some of the underlying causes of their increased risk of infectious morbidity might be altered immunity. And there have been quite a few studies demonstrating that they have a sort of an innate immune activation. They definitely have altered humoral responses to vaccination, and they inherit very different both quantities and quality of maternal antibody transplacentally. And some studies have also suggested impaired antigen-specific T-cell responses, including to BCG. And BCG is the vaccine we use a lot in our lab because it's a prototypical neonatal T-cell vaccine that induces really nice T-cell responses in infants. So here's some background data also from our group, published by my colleague Sonwa Zanibe, where we took infants from an observational cohort and did multi-parameter flow cytometry, or actually CyTOF. We looked at various cell populations, and here I'm just showing you the CD4 T cell populations. You can see the ontogeny in HIV-exposed infants is very different to that of unexposed infants. And we look at using unsupervised clustering, the cell types that are driving these differences. In summary, I have here that in HIV unexposed infants, there seemed to be an enrichment of activated CD4 T cells that are perforin expressing or terminally differentiated. But in HIV unexposed infants, there's an enrichment or Th2 skewing of CD4 T cells. And what's more, ourselves and other people, this is a study from Nigeria, have found that there's alterations in the gut microbiome in infants exposed to HIV. Particularly even in those that are breastfed, because of course feeding makes a huge difference in gut microbiome. But amongst breastfed infants, HIV exposed had significantly lower alpha diversity, clustering of their community composition in a PCA, and a lower relative abundance of bifidobacterium at 6 months of age. So we'd been thinking for a while, is the gut microbiota what underlies these immunological differences and potentially also even neurodevelopmental differences in HIV-exposed infants? So I've been lucky enough to be recruiting a cohort from Site B Khayelitsha in Cape Town for the past 11 years now, I think. It's an observational study. We've recruited now over 1,000 infants that are born to mothers with and without HIV. Site B, for those who don't know, is a formerly informal settlement with high rates of poverty and HIV. And for the purposes of this analysis, we've included only vaginal deliveries and infants that were breastfed. The cohort is designed as follows. We collect stool at multiple time points from both the mom and the baby, and blood. BCG vaccination is given within the first couple of days of life. And we do a whole bunch of stuff with that blood, including measuring BCG, Th1 T cell immunogenicity. We do this at various time points in the baby's life by collecting blood and placing it immediately ex vivo into an incubator at the clinic with BCG, media alone, and a positive control, incubated for 12 hours, and then store the cells, and at some point we defrost and measure interferon gamma, TNF, and IL-2 being made by these cells. And here you can see in the bottom left, the total net CD4 Th1 cytokine responses versus the number of infants. And there's a huge range in the proportion of cells that are making Th1 cytokines after this 12-hour incubation, ranging somewhere from 0 to all the way to almost 5% of the cells. And for those of you, so we've binarized these responses into low responders, and you can see an example here, and high responders around the median. And for those of you who believe in COMPASS polyfunctionality scores, there's a very strong linear relationship between the COMPASS polyfunctional score and this net CD4 Th1 T cell cytokine response. So what we did in these infants is look at their gut microbiome around the time that they were born, or around the time that BCG is administered. So in the first couple of days of life, and there weren't any huge differences in alpha and beta diversity between the communities of infants who are high versus low responders. But you can see that these are the significantly differentially abundant organisms. And there's some organisms that are enriched in high responders, and a lot of organisms that are enriched in low responders. So we selected Bifidobacterium longa, subspecies infantis, as a sort of a representative high responder bacterium, and Bacteroides theta iota micron as the low responder example. And I'll call that B theta from now on because it's rather a long name. And we decided we wanted to see if this association in human infants may actually translate into causality in a germ-free mice. So we performed monocolonizations. And this is already published, but I'm going to tell you where we're taking this from now. So real quickly, we monocolonized the mice with B theta, B infantis that was live or heat killed at day 7 and 10 of life, vaccinated them at day 11, and harvested their spleens for tetramer staining for TB10.4, which is a BCG antigen, at day 39. And you can see an example of the B theta response of activated TB10.4 specific cells and a B infantis response. And when we summarized these, we found that B infantis was, mice that received B infantis had significantly higher TB10.4 specific T cells compared to B theta, but also compared to heat-killed B infantis. We then went on and did this in SPF mice, so mice that did have a microbiome, because obviously if we're ever going to use B infantis as a biotherapeutic, we're going to give it to infants with a gut microbiome and not germ-free ones. So we did the same experiment where we monocolonized at day 7 and day 10, vaccinated and harvested, and stained for TB10.4 tetramers. And here you can see, and sorry, this is a bit shifted, that like the germ-free mice, the B infantis gavage mice had significantly higher T cell responses to BCG compared to B theta or PBS. But the heat-killed B infantis mice also had a really significantly higher response, suggesting that if you have a native gut microbiota, you could supplement just with a heat-killed B infantis to recapitulate the phenotype, suggesting that we could even give we don't need necessarily give live B infantis as a supplement. We tried to get a little bit at mechanism. So we did the same experiment, but at day 14, we harvested the colons of the mice. And you can see the colonic transcriptome of the mice was very, very significantly clustered according to treatment with B infantis and heat-killed B infantis. And this was in the SPF mice, clustering together. And separately from from B theta and PBS. And when we looked at the transcriptome, we see genes such as MADCAM and other sort of cellular matrix genes enriched in B infantis gavaged mice, but genes associated with inflammation in the B theta gavaged mice. And when you look at the KIG pathways that were enriched in B infantis, there were extracellular matrix receptor interaction and focal adhesion, whereas in B theta, they were RIG and NOD-like receptor signaling and IL-17 signaling, so very much inflamed in B theta. And this was very consistent with another study that was published meanwhile, whilst we were doing these experiments, where they randomized, it was called the imprint study, published in Cell. They randomized 60 breastfed infants to EBC001, which is a type of B infantis, versus a placebo. And you can see, really, this is day 6, there's no real change, but at day 60, you can see in the fecal, in the feces, an upregulation of interferon beta in the probiotic receiving infants, and a downregulation of Th2 and Th17 related cytokines. And when they looked at the fecal metabolites, the metabolite that was most enriched in infants who received B. infantis was this indole lactic acid, or ILA. And they then took fecal water, exposed PBMCs to it, and found that fecal water prevented Th17 and Th2 cell polarization in PBMCs. And this was their model, that high HMO utilizing capability given by B. infantis, but potentially by other bugs, increases indole 3 lactic acid, which causes a sort of a lack of intestinal inflammation and a Th1 skewing. So that could be very much what was going on for us. So in summary, and Joe, will you just wave to me when it's time for me to wrap up? Thanks. We found that B. longum, subspecies infantis, influences T-cell immunogenicity of vaccines, that it has the potential as a biotherapeutic or probiotic to improve Th1 responses in neonates. And that's something we might want to do for a TB vaccine, or maybe even potentially an HIV vaccine, where we want good immunity very early in life and T-cell immunity. And we don't completely understand the mechanism, but it's possibly due to lower mucosal inflammation and hence maybe increased indole 3 lactic acid or other metabolites, increased barrier function, and Th1 skewing. So that's all published. But since then, we've been lucky enough to get funding to take this further. And so we're conducting a trial called BIFID, which stands for B. infantis, for infant immune development. It's a randomized, placebo-controlled, quadruple-blinded trial of B. infantis strain Rossell 33, which is donated by Lalemant, and they've been absolutely wonderful to work with. We're giving them 8 billion CFUs per day by mouth for the first 4 weeks of life. And we're doing it in 200 infants, randomized placebo to active biotherapeutic, and they're all HIV exposed. So aim 1 was to compare gut microbial structure and function longitudinally in these infants, and to evaluate associations with the stool metabolome. Aim 2 is to compare gut mucosal integrity and immune ontogeny in these infants. And finally, aim 3, which is really my primary aim, or probably the primary aim of the trial, is to compare the T-cell responsiveness to BCG vaccination, and then other clinical outcomes such as linear growth. And then since then, our colleague, Dr. Happel, has received K43 funding to also assess neurodevelopment in the infants at 9 months and 2 years of age. I've got a great team of collaborators, including Anna Happel at UCT, Clive Gray at Stellenbosch, Sean Gibbons, who's at Institute for Systems Biology here in Seattle, and Donald Nyanga, who's now actually just gone to Rutgers to take a faculty position, but he was at Seattle Children's with me. The study schema is that we randomize babies within the first 3 days of life to, as I mentioned, 28 days of daily B. infantis, and we've chosen to give them live B. infantis. It has a really good shelf life, but I'm not actually sure that's really necessary. We're following the babies. Initially, we were following them to 36 weeks of life, but since Anna has the funding to do the 2-year neurodevelopment until 104 weeks of life, we're collecting, again, the same large amount of stool and blood and maternal samples too, including breast milk. And we'll be doing, as I mentioned, we'll be ruling out HIV infection, measuring vaccine immunogenicity, looking at immune ontogeny, mucosal integrity, and performing shotgun metagenomics and metabolomics on the stool, and we're collecting maternal samples just to see, how much does a maternal phenotype affect what we see in the infant, despite supplementation. Okay, so this is our study poster. Are you pregnant? Are you interested in potentially boosting your baby's immunity and health? Please contact Busi Tahengi. And we began enrollment in August 2023 at Site B, and now we've added Guguletu, and enrollment is going so fast, the pharmacies asked us to slow down. We, I think, have 88 mother-infant pairs enrolled as of Friday, and 13 infants have completed the study. And so please watch this space. I'd love to give you some results at number 5 of the African Microbiome Day. I think we'll need that much time to analyze all the results. But I'd like to thank my colleagues, particularly Donald Nyangahu, who performed most of the in-mouse experiments, but also my lab and clinic at UCT, who both collected all the samples that from human infants and is helping with the BIFID trial. I'd like to thank Lalimand for their very kind collaboration, people in Seattle who've collaborated with me, and at UCT and Stellenbosch, our participants, our funders, and the Department of Health for allowing us access to their clinics. And with that, I'll stop sharing, and I see there's already one question.
So thanks, Heather. That was very, very exciting. I've got a couple of questions, but I'm going to go to Philip's question in the chat, and he wanted to know, are HIV exposed and infected children in South Africa given prophylactic antibiotics? And if so, what would the impact be on the microbiome?
Yeah, it's a great question. We anticipated and we justified very well in the grant, I think, that this would be of benefit despite Bactrim prophylaxis or cotrimoxazole. However, South Africa has changed their guidelines since we started the trial, and we aren't giving infants exposed to HIV cotrimoxazole anymore. So that'll remove that very interesting confounder. But thanks for a great question.
Thanks, Heather. So in the interest of time, we're going to move on to the next presentation, which is a short presentation. And maybe at the end of the session, if we've got time left over, there are questions still popping up for Heather. We can go back to our speakers and ask, have a bit of a Q&A. With that, we've got flash presentation from Caroline Musimi. I apologize if I've pronounced that wrong, but we look forward to the flash presentation. And Caroline, please be available if there are any questions for your talk.
Hello everybody, my name is Caroline Musime, and I'll be presenting my research on investigating gut microbiome diversity among children infected with schistosomiasis in the endemic region of Albert Nile in Pakwach district in Uganda. The schistosome species, also known as blood flukes, cause schistosomiasis, that is commonly known as bilharzia. It mostly affects 10 to 20-year-olds and is known to have caused more than 290 million infections worldwide, with an annual death rate of approximately 280,000. These intestinal helminth parasites have been found to live in proximity with gut bacteria. However, their interactions have not been fully investigated. They can cause changes to the gut microbiome composition that is observed in this image on the right, where we see that bacterial diversity greatly reduces when there is worm infection in the ileum. We aimed to investigate the gut microbiome diversity's effect on schistosomiasis infection intensity among the children. This was a retrospective analytical study where 10 to 15-year-old school children were recruited, both those infected and uninfected with S-Mansoni worms. The study site shown here has 6 sites where samples were collected from, with Alui being the farthest from the shore and having the most number of controls, and areas that were closer to the shore having higher numbers of infection, with Nyakagei having the highest number of high infection intensity. The infection intensity was measured using the Cato-Katz method. Eggs per gram were determined through microscopic detection in the fecal samples. The parameters used were control, which had eggs per gram as 0, then low infection with 1 to 100 eggs per gram, and finally high infection with eggs per gram as more than 100. A total of 138 archive fecal samples were used for this study. DNA extraction was done, followed by 16S rRNA library prep of the V3, V4 region. This was followed by sequencing on the DNB platform, and the results obtained were used for taxonomic classification and abundance enrichment analysis. Figure A shows us the number of operational taxonomic units that were identified, and we had a total of 1,452 OTUs, with a number of them being shared amongst the three groups. And then figure B is a plot of the principal coordinate analysis of all the three groups, where we can see that there was no clear clustering within the three groups by infection intensity. Plot A here shows the distinct differences that were observed in the most abundant phyla that were more than 90% of the total phyla within the samples, and these were the four highlighted here: Firmicutes, the Bacteroidota, the Spirocheta, and the Proteobacteria. Plots B and C were the alpha and beta diversity measures within the three groups. These highlighted phylum and classes were the taxa that had the most and highest abundance in regards to high KK intensity observed within the samples. Levse analysis was then done to determine the genera that were differentially enriched within the samples, and these are the ones that are highlighted. And Prevotella was found to be the most abundant, which is known to play a role in glucose metabolism. In conclusion, there was dysbiosis that was observed within the different infection intensities, and the enriched genera are involved in metabolic functions, and therefore they have biological implications. The hymen indices reflected species diversity and richness of the microbiome communities within the three groups, and this data therefore suggests that the gut microbiome could be an important variable in this disease progression in children. Thank you for listening.
Thanks, Caroline. That was a very interesting talk, and schistosomiasis is so under research. So looking at the intersection between the microbiome and schisto is extremely well placed in this conference. I don't see any questions coming up in the chat, but I had a really interesting, difficult one, and I get asked this all the time, and I can't answer absolutely. But I was wondering if you could speculate about like the causal pathways. So if you've got an association between a dysbiosis and schisto, if my memory serves me well, you get those sandy patches, and you get quite a lot of irritation in the epithelia. Like, in terms of plausible pathways, what are your thoughts on whether the dysbiosis is caused by the sandy patch and the schisto, or vice versa, where, you know, what do you think the relationship is?
Can you hear me?
Yes, I can. Thank you.
Yes, that's a tricky question. But I think that dysbiosis, I think it's caused by a number of factors that just interplay within each other. I don't think it's just one thing. That would be my guess for now. Yeah.
Yeah. I read quite an interesting paper on the intersectionality of papillomavirus infection and BV, and you know, historically people have thought that having BV increases your risk for HPV infection, but there was a recent paper in can't remember, Science Communication, looking at the fact that an HPV infected tissue could also change how lactobacilli thrive in the vagina. And that's, you know, it comes back to, we tend to look at the association, but actually understanding the biology underneath that is really interesting. But thank you very much for that presentation and for that research.
Thank you.
So we are perfectly on time, I'm very happy to say, and thank you both to Heather and Caroline for keeping us on time. But I would like to introduce the next speaker for this session is the amazing Dr. Disebo Potlani from Caprisa. And I have come to work with Dr. Potlani quite closely in recent months. Dr. Potlani is site director and a clinician from Caprisa at their Vulindlela Research Clinic in KwaZulu-Natal, South Africa. She is leading the study that you are going to be hearing about today, which looks at a new product aimed to shift the vaginal microbiome in women with bacterial vaginosis. And this trial has gone through SAPRA, and we're all waiting with bated breath to see the outcome of the study. And I'm really looking forward to your talk, Disebo. So I'm hoping you are able to share your slides.
Good day, everyone. Thanks for that very kind introduction, Joanne. The amazing Dr. Potlani is one that I've not heard yet, but thank you so much. I'll just go ahead and try and share. Please let me know if we are in presenter mode, presentation mode. Awesome. Thank you so much. So as Joanne has stated, I'm presenting on this very exciting project that we're doing that looks at a multi-strain Alcrospathes live therapeutic product that we're hoping will be able to modify the vaginal microbiome in women with BV. So I am presenting on behalf of the Vaginal Microbiome Research Consortium's clinical group, and I co-lead the project with Dr. Caroline Mitchell, who is based in Boston. So this is just a brief overview of my presentation for today, and we'll get right into the need to address vaginal dysbiosis. So as we've heard from earlier presentations, that lactobacillus-dominated vaginal microbiome, and more specifically, one that is dominated by Lactobacillus crispatus, is a hallmark of a healthy vagina and female genital tract. We've heard earlier that BV is characterized by an increase in diversity and quantities of species in the genital tract and the absence of Lactobacillus colonization. BV affects approximately 30% of women worldwide. And as one of our presenters mentioned, that women of African descent are actually disproportionately affected, as they are more likely or more commonly have these diverse vaginal microbiota when compared to European and Asian women. So BV is a risk factor for multiple adverse reproductive health outcomes, including HIV infection, cervical dysplasia, and STIs infection, and and we know that from data, this inflammation that is associated with these STIs and with BV increases the risk of HIV infection, but more specifically, it facilitates the acquisition of less infectious HIV variants, which is extremely important in HIV hyperendemic regions such as Eastern and Southern Africa, where we see that also the prevalence of BV is highest. So we know that BV is treated with oral and topical antibiotics, mainly metronidazole and also clindamycin. And we see that there are high recurrence rates of BV following treatment with these antibiotics. This is data that was generated from a study called Caprisa OH3 study, where women that had BV had their vaginal communities or the community state types characterized at baseline. they had their, they were treated with metronidazole, and then the CSTs were characterized again 6 weeks afterwards and at 12 weeks following treatment. We see in this data, I don't know if you can see my arrow, but a lot of women have vaginal communities that are dominated by L. innes, which is represented by these yellow bars, rather than crispatus after having been treated with antibiotics. At the 12-week time point, we see even more women that have this L. innes dominance. And we know that innes is not as beneficial in preventing adverse reproductive outcome as crispatus is. So this supports the information that we have that treatment with antibiotics does not promote L. crispatus dominance, and rather women will come up with this L. innes dominance following treatment with antibiotics. This also means that we still need an intervention that can promote L. crispatus dominance in women that have dysbiosis. So what are some of the strategies to promote L. crispatus dominance? We've seen various commercial probiotics that are targeted for vaginal health, but these products don't contain the lactobacillus strains that are present in the female genital tract. They also show minimal to no benefit after being used. A possible strategy would be to inoculate or deliver intravaginal exogenous L. crispatus strains in the form of a live biotherapeutic product. And this strategy it has been done in a few studies, but it was also done in the Lactin V study that used a single strain of L. crispatus. We see from the data from the Lactin V study that although BV recurrence was lower in the Lactin V arm compared to placebo, these recurrence rates were still high and it increased over time. So that means we still need to find a product that can durably shift the vaginal microbiome to one that is dominated with L. crispatus. So on this slide, we're seeing data that was generated by a group at the University of Maryland led by Jacques Ravel. And they looked at the bacterial genes in women that had these stable L. crispatus dominated vaginal communities. And they found that the number of genes, unique genes that are present in the metagenome are much higher than the number of genes that are present in any single isolate of L. crispatus. So that means that when you look at any single isolate, there are a number of metagenome genes that are unaccounted for by any single L. crispatus strain that is present in these women. On the far right-hand side, we see that when you start combining strains of L. crispatus, you get this increase in the genetic coverage. And when you get to about these 6 strains, you get to reach this plateau phase where there is still an increase in genetic coverage, but not one that is as remarkably demonstrated from 1 to 6 strains combined. So various people looked into their cohorts. Various scientists in the Vaginal Microbiome Research Consortiums went back to their cohorts, and they went to identify these women that had women in their cohorts that had these stable L. crispatus dominated communities. And from these women, they isolated those specific strains and combined these strains together. So this is data from a CAPRISA study called CAPRISA 084, which was a PrEP demonstration study. But a similar exercise was also done at UCT, at the FRESH site in Durban, and also in the US, which led to us having this combination of 15 genetically unique L. crispatus strains that were isolated from women in South Africa and in the US. These strains were then ran through an in silico algorithm, and we identified 6 of these strains that were able to produce about an 80% genetic coverage when doing metagenomic studies. And these 2 combinations are what leads to the two products that we'll be using in the Vibrant trial. So a 6-strain combination called LC106 and a 15-strain combination called LC115. So the Vibrant trial, or the Vaginal Live Biotherapeutic Randomized Trial, is a phase 1 first-in-human trial that aims to assess the safety, tolerability, and colonization kinetics of LC106 and LC115. Our objectives include the safety and colonization kinetics, the capacity of the L. crispatus strains that are contained in the products to establish colonization in women from these two geographical cohorts, and also to identify optimal dosing frequency and duration that we're hoping to use in future studies. The trial is being conducted at 2 clinical sites, the Massachusetts General Hospital in Boston and the Caprisa Vulindlela site in KwaZulu-Natal, South Africa. This is just a brief overview of the people that we're including in this study, and that's 18 to 40-year-old premenopausal, non-pregnant, HIV uninfected women that haven't recently used antibiotics, pre-pro symbiotics, that are not that are refraining from vaginal cleansing practices or the use of any vaginally inserted products. This is just also a summary of some of the tests that we'll be doing and some of the data that we're collecting on CRFs, or case report forms. A quick schema of the study. So following successful screening, women will be randomized, or are being randomized, into one of 5 arms. All participants in the study receive a 7-day course of oral metronidazole per the local standard of care. In the first arm, participants will receive a 7-day course of a vaginally inserted placebo tablet. In the second arm, 7 days of LC106. In the third arm, 3 days of active 106 and 4 days of placebo. In the fourth arm, 7 days of 106 that overlaps with the antibiotic course. And in the last arm, 7 days of daily LC115. We then will go into an off-product follow-up and an extended follow-up that will hopefully be able to show durability of the product. In parallel with this clinical trial, we're also conducting a qualitative ancillary study that aims to understand user experiences in women that are using the product as part of the clinical trial. This is just a brief overview of our study outcomes. So our primary outcomes include safety that we'll assess by the rate of adverse events and serious adverse events, colonization with L. crispatus strains that are in the product over the first 5 weeks of follow-up. And this is a brief list of our secondary outcomes, which includes BV recurrence, product acceptability, tolerability, a change in diversity and composition of vaginal microbiome, and we're also hoping to compare effects between the US and the South African side. So in conclusion, particularly for me at the Vulindlela site, I'm hoping that we'll be able to reduce the risk of HIV infection, but with this product, we also have the potential to reduce other adverse reproductive health outcomes in women. It's a first-in-human study that is, the first step along the road to, shift the microbiome towards L. crispatus dominance. Our long-term goal in the project is to identify an intervention that can prevent incident BV in people at risk, and we hope to be able to create a durable and predictable shift of the vaginal community to one of L. crispatus dominance. So these are some of the institutions represented within the VMRC, and I really should have everybody's names because this is a massive effort. But yeah, just like to thank everybody that is involved in the Vibrant Trial and project, and obviously our funder, which is the Gates Foundation. Thank you so much, Joanne. I'll stop sharing.
Thank you so much, Desevan. As I said, we are so excited to see the results of this. And I do know how much work has gone in the background in working to get the right woman into the trial. I am just quickly scanning the questions. I was wondering whether you could comment, because I've got insider knowledge, as you know, but one of the enrollment criteria for the study, because it went through the US FDA, was symptomatic women with bacterial vaginosis. And I know that has been incredibly difficult to find in South Africa, and particularly in the site that you are based at. Can you comment on maybe why it's so difficult and why there is a difference in phenotype in bacterial vaginosis presentation in different cohorts of women?
So yes, one of the criteria that the FDA wanted us to include was the AMSEL criteria. And we have found that women that have BV by Nugent score often have, low AMSEL criteria, or they don't meet the 3 out of 4 AMSEL criteria that we had in the study. We've also looked at other data in other cohorts in Africa that showed that, high Nugent scores don't necessarily always correlate with AMSEL positivity. And that actually led to us pleading our case to SAPRA to not include AMSEL as an inclusion criterion for women that are enrolled in South Africa. So actually that amendment has just been approved by SAPRA and our local ethics committee. And what that does is that it allows us to have a broader set of women and to not exclude women that actually need this intervention. A lot of BV is asymptomatic in African cohorts, but the sequelae or the consequences of this dysbiotic vaginal community still remain. So we really need to be making sure that we're not excluding people that could benefit from not only study participation, but also this product, should it be successful.
And as a follow-on to that question, as you know, I'm a laboratory scientist, and I feel that asymptomatic BV is as bad as symptomatic BV, even if the overt AMSEL criteria clinical symptoms are not clearly evident. But there's been a lot of pushback in the field for treating with antibiotics asymptomatic BV. Because it's not a clinical condition and therefore doesn't warrant antibiotics. Can you comment on, what your thoughts are on whether SAPRA had any concerns about using antibiotics for asymptomatic BV?
So no, SAPRA didn't raise any concerns with using antibiotics in asymptomatic, and neither did the ethics committee. And I agree with you. The sequelae of these dysbiotic vaginal communities still remain, as I said earlier. I fully understand, the reluctance with using antibiotics in women that are asymptomatic, and it speaks largely to rational and responsible use of antibiotics. We also see, such high prevalence rates of women that have high pH, women that have high Nugent scores, and we would essentially be treating masses and masses of women with antibiotics repeatedly. And from a clinical standpoint, that is not necessarily, the best approach, which is why we need to be looking at other interventions that will deal with dysbiosis, but not burden our participants with these repeated courses of antibiotics, which obviously will drive resistance and side effects and all other adverse events following that. So I think, I'm glad the live biotherapeutic space is growing, because we do need interventions even in women that are asymptomatic.
Thank you so much, Disebo, for agreeing to participate in our third event of this nature. And we really appreciate you putting the time and effort into giving us a talk.
Thank you. I look forward to presenting results this time next year.
Yes, so do we.
Thanks, Joanne.
So I'm the exiting chair, and we are now heading into a session with Dr. Vakaria Oderin from Wits University, from the Sydney Brenner Institute. As you know, I don't think she needs much of an introduction. She is the standing chair of this third event, and is also one of the most passionate people about raising our game and appreciation for this field. Voke, as we all know her, has been very influential in the field. And I've just posted a link to a really, really important commentary she authored in Nature, looking at how we can do microbiome research more equitably across Africa and with our global partners. And I'm really excited, and I'm hoping everyone's enthusiastic to hear the message that she's going to be bringing to us over the next 10 minutes. But I just wanted to, on behalf of everyone, thank Voke for her undying, passion for this and for moving this successful microbiome research day forward each year. So over to you, Voke.
Thanks, Joanne. That's very kind. Great. Can you all see my slides? Excellent. Thank you so much. Thank you so much, Joanne. Thank you so much, everyone, for just being present and just participating. To the invited speakers, thank you so much. I'm sure I'm going to do a thank you again at the end, but just saying thank you ahead of time. And thanks to all the those who submitted the abstracts, and just for those that are attending, we're so grateful. You make this very special. You make this, make us come back to doing this every year, just your participation. But you realize that every year we come back and do the same thing, and with, different sets of speakers, but there is a goal here. And that vision has always been to have something more collaborative, something more that we can all actually work together, do something together that would be tangible, go with our numbers and the collaborative efforts we have to influence the space of microbiome research, both in Africa and in the global stage. So I'd just like to give a bit of a background just very quickly. So just so you know that this meeting is being organized by the African Microbiome Special Interest Group and the H3Africa Microbiome Task Force, like I mentioned earlier. So the first meeting of the H3 Microbiome Task Force was, came together at the 14th H3Africa Consortium meeting in Ghana in September 2019. So we've been around for a while, crawling sometimes, but jumping sometimes and flying other times, but we've been around. The general idea was to build capacity in microbiome research on the continent amongst the H3Africa projects that were foraying into this field, because it was relatively not very well researched at that time, and to create avenues for networking and collaboration, like I said, to accelerate microbiome research on the continent, and also to improve representation of African microbiome data samples in the global microbiome space. But very important, they would do all of these to improve the health of and just the general life outcome, health outcomes of the African population, and also in extension to the global population as well. But we're also aware, just to say, that the funding cycle for H3Africa ended in June 2024 officially, but the task force continues. But in that line, last year, about a year ago, well, a little over a year ago, in June 2023, we started meeting. We expanded the task force to include everybody working on microbiome research, intending to work on microbiome research on African populations. So we had a first meeting on June 2023. I'm just so thankful for all those that have just been consistent and just supportive of the initiative. This was because in previous meetings like this, we've had calls for something more inclusive, something more collaborative, something on a broader network, not just the task force. So we made that open, and it's still open for everybody to join. That meets the criteria of working on African microbiome research or interested in working with African microbiome populations. So the main thing, the main idea was to build on the efforts of the task force and expand the network, obviously going outside of H3Africa projects and just encompassing, just having everybody doing something with microbiome research on the continent come together. What it allows for us to do is that we have a bigger network and we're able to identify synergies across research efforts within the continent, because you realize that, especially from these meetings, thanks to these meetings, that there are several small projects going on in little silos and people doing similar things or related things that could actually work together and have a bigger impact. So that's one of the things we hope to accomplish. So in line with that, we've always had this vision, and all of this is leading towards that. We still have a task force, but we're hoping to have something more collaborative, an African Microbiome Consortium. You may have seen a QR code at the beginning and at some point during the break, maybe talking about with a survey for African microbiome projects or studies, because we're looking to gather all of this information to help inform us and just help us plan better as we take this tangible stride towards coming up with an African Microbiome Consortium. Just looking at different consortia over the years, even just looking at the H3Africa Consortium, I thought about if people came together, thought of what would be amazing for Africa in terms of genomics. And today we hear about the H3Africa Consortium in just about every part of Africa, when you talk about human genetics, and just to see how much that has improved human genomics in the knowledge of African human genomics globally as well. So we have that vision as well for the African Microbiome Consortium. It's not a one-person project, not a one-lab project, but we want to do this together and make sure that our impact is felt for us in Africa and also on the global stage. I've just copied and pasted the vision statement here. People have been working behind the scenes just to come up with our vision statement based on the activities so far and just the general terms of reference of the African Microbiome Special Interest Group and the task force. The idea is to position Africa as a global leader in microbiome research and innovation. I think that's a shared interest for all of us here. To harness the power of scientific collaboration, a very big deal, because we know that together we can, to address the continent's unique health, agricultural, and environmental challenges. We definitely envision a future where African-led collaborative microbiome research drives sustainable development, improves public health, and enhances food security, all the while protecting the continent's rich biodiversity and ecosystems. This is a dream we have, and I'm sure that even as we do our different research, our different projects, it all kind of feeds into this overall vision. And I'm hoping that we all buy into this dream. This is not a pipeline dream, I promise. It's something that is achievable, and we can already see tangible steps towards this being accomplished. And the mission statement, just to make it a bit formal as well, is like to drive impactful microbiome research across Africa, fostering collaboration. As you might have noticed, collaboration is a big deal within African, with African, the global North as well, enhancing local research capacity. You could see from the different presentations you saw, both the recorded ones and even from the invited ones, that there's a varying level of expertise that could definitely be worked on to give us better outputs for our projects and our efforts. We're doing great, but there's always room for improvement. I would like to build more on what we have. We'd like to develop innovative and tailored solutions for African problems, so African challenges as well. We're committed to ethical practices, inclusivity, sustainability, standardization, and leadership. To this end, I haven't included that here. We have 2 publications that came out this year. One encouraging standardization in microbiome data collection and just microbiome data practices across the continent. That was led by Lyndon Zass in collaboration with the H3 Bionet. And we also came up with the article, the comment article, Johan just put in the chat a short while ago as well. That was something we did collaboratively as a group. So it wasn't my individual effort. I mean, I've led it, but it was like a collaborative effort, definitely a team effort. So we hope that by facilitating access to resources and promoting knowledge sharing, will empower African researchers, improve public health, again, boost agricultural productivity, safeguard the environment, it's a big deal, and contribute to sustainable development across the continent. Our ultimate goal is to create lasting positive change that benefit African communities and strengthens the global research community. To this end, again, I say together we can. Please feel free to scan this barcode. It's going to feature several times throughout it's been featuring since the beginning of the event. We're serving African microbiome studies and projects. Would like to know more about what you do. If you're interested, please go ahead and scan out. If you don't have a project right now, just reach out to us if you're interested in this initiative. We can see that from when we started in 2019 till now, there's been a lot of more projects, a lot more papers on the African microbiome, which we're very thankful for. And we are looking forward to the next step, which I think is the African Microbiome Consortium. And we invite you to join us along this process. To acknowledge, in terms of acknowledgement, I'd like to thank the groups, the African Microbiome Special Interest Group, the African Microbiome Task Force, and also, like I said, the H3Africa Microbiome Task Force. The whole H3Africa funding, it's come to an end now, but we're very grateful to the NIH, the Wellcome Trust, H3Africa, we're very thankful for the opportunity as well, and to Science for Africa. We look forward to the next steps. And I hope I have done 10 minutes, not more than 10 minutes. Thank you so much. And that is the end of my presentation. I'll stop sharing my slides now to get into my other role, which is to introduce our keynote speakers. Please feel free to pop in questions into the chat and just get in touch, engage. We're very excited to talk some more about this, but we don't have limited time here. So, so thanks everyone. With that, I'd like to very, very happily introduce our keynote speakers. We have a joint keynote from very, very special people to me. They are great mentors. We've done this as a joint keynote because it is really what we hope to see going forward in the African microbiome research space, collaborative efforts. I believe this is an excellent example of very productive, equitable, amazing collaborative efforts within South Africa and the US. I'll very happily introduce Professor Ami Batts and Professor Scott Hazelhurst. I know they wouldn't like a lot of introductions, but I'll go ahead and just give something brief anyway. Prof. Ami Batts is dedicated to inspecting, characterizing, and dissecting the human microbiome interface. She does this excellently well, very humble. She doesn't talk much about her accomplishments, but she's received several awards as well. She's from Stanford University. Oh, sorry, I didn't say that, but she's from Stanford University and has an amazing lab there. She's a recipient of several awards, like I said, including the Microbiome Data Prize from the American Society of Microbiology. This was this year. And she's a professor of medicine, hematology, and of genetics at Stanford. And I'll just quickly switch gears to Professor Scott Hazelhurst. He's a professor of bioinformatics in the School of Electrical and Information Engineering and a senior scientist at the Sinibrenner Institute for Molecular Bioscience. He's the PI of the Madiba Research Hub, which is the Multi-Morbidity in Africa Digital Innovation, Visualization and Application Research Hub. And it's part of the Data Science Innovation for Health in Africa initiative by the US NIH. So without taking much more of your time, because I want us to get straight into this, I'd like to just invite Professor Ami Batts and Professor Scott Hazelhurst, amazing mentors to me, and I'm sure that we've heard about them severally, to talk about expanding the human gut microbiome atlas of Africa. Please take it away. Thank you so much. Scott, you're muted.
Yeah. Sorry, 1 minute. Sorry, problem of a new computer. And Sorry, Volker, I'm sorry to do this to you, but I've got to disappear for a minute and come back.
That's okay, Scott. And just to add while Scott is getting a setup, please feel free to add in any comments or questions in the chat. Thanks for making that a very active means of communication so far. So let's just continue using that as well. Thank you.
Sorry about that, Volker. Problem of a new computer. It wouldn't let me share my screen. But I think it's working now. Can you see my my screen?
Yes, it can.
Great.
That's
Okay, thank you very much for the invitation. It's really amazing being here. It's fantastic to see how this day has grown over the last few years. So congratulations to you and the whole team for making this happen. I thought the standard of the presentations was excellent. So it was very exciting being here. Ami and I also are very excited about being here because this is work that we first started talking about 8 or 9 years ago, and we started seriously working on this about 7 years ago. And it took a long time to happen with a lot of people participating. So as you said, this is a team effort. It's a joint collaboration between Ami's lab at Stanford and the AWIGEN collaboration. So we're going to do this in 3 parts. I'm going to start by just setting the context for the work that we've done and a short motivation. Then Ami will present our initial results, and then I'll end by reflecting on the way we went about the work, both the successes and perhaps some of the mistakes that we made. So I think that I don't need to belabor the motivation at this presentation, given all the talks we've had. Obviously, the microbiome has an important contribution to human health. And despite all the excellent work that's being presented here and that's happened in the past, we know that despite the very significant diversity in Africa, Africa is also underrepresented by any metric in microbiome and health and genetics research. So we were fortunate because we were collaborating as part of AWIGEN that we felt we could make a real contribution to the microbiome and health. So the AWIGEN Collaborative Centers, one of the H3Africa Collaborative Centers, it's led by my colleague, Michelle Ramsey at Wits University at the Sydney Brenner Institute. And it's a collaboration with a number of other institutes. We have 6 sites in 4 African countries: University of Limpopo, African Population Health Research Center in Nairobi, the Navrongo Health Research Center in Ghana, and the Clinical Research Unit of Nenoro. So this is designed to be a longitudinal study. We've done 2 phases. We recruited 12,000 people in phase 1, and 7,200 were followed up in phase 2, and we hope to do phase 3 in a few years' time. So in all of AWIGEN, which is much more than the microbiome project, we have very rich data sets that are exploring a range of scientific and health questions. What's very exciting about AWIGEN is that by being set in different countries, in places that are at different stages of epidemiological transition, we are able to explore some of the socioeconomic diversity and the genetic diversity and other types of diversity that we find in Africa. We think that's very important because often there's a flattening of what people see as Africa, as if it were one place, if it were one country even. So I think being situated in AWIGEN allows us to explore the role of the microbiome in different stages of epidemiological transition. So the broader AWIGEN project has a number of themes, and I'm not going to go through them all. You can see on the left-hand side the themes that are being explored by AWIGEN, and on the right-hand side, some of the phenotype data that has been collected both in phase 1 and in phase 2 of AWIGEN. So the microbiome project, which required several villages to make happen, is a sub-study of AWIGEN. We recruited 1,800 of the women in the broader AWIGEN study. That's about 40% of all the women. So because it's nested in AWIGEN, we have very rich phenotype data. We now have rich microbiome data, and we also have the human genotype data that was collected in the first phase of the AWIGEN project. It was a long time coming, as I said at the beginning, and it wasn't just about going into the field, collecting data and doing bioinformatics analysis. It was about working with different people. For example, we ran in collaboration with H3A Bionet, we ran various teaching and training workshops in bioinformatics analysis. We also did community engagement at various phases of the project. So the microbiome project itself is quite ambitious. Maybe we think at this stage too ambitious. So we've got lots of research questions we want to explore. Ami is going to present our first results from our first analyses. And broadly, I've given it the very unglamorous title of what's there. It's much more than that. It's about what drives the diversity of the microbiome at the sites that we did the work in. We try to understand what the importance of the microbiome and what it does, the novelty that we found. And then we'll be looking at the impact of health on the microbiome and vice versa. And in particular, we'll be looking at menopause because there's a companion study in AWIGEN looking at menopause. And then because we've got the human genetics data in AWIGEN, we'll be looking at the three-way relationship between the microbiome, health, and human genetics. So that takes me to the end of my first part, and I'm going to stop sharing, and Ami is going to take over, and she's going to present the part that we call expanding the African gut microbiome.
All right, thank you so much, Scott, for that great introduction. And thank you so much, Voke and colleagues, for your time today, and Voke and the whole team that organized this for your tireless dedication to making sure that we can all share our learnings and our advances. So as Scott mentioned, I'm going to be giving us a little bit of a highlight of findings from this first round of analysis. Just to catch everybody up to speed, you all know this very well, you're researchers in this space, but I'll just remind us that we studied the microbiome by collecting samples, extracting DNA, sequencing reads, and then doing things like calculating the abundance of different organisms that are present. Obviously, embedded within this are a lot of choices that one can make. And for those who are interested, Yishai Pinto, a postdoctoral fellow of mine, and I recently wrote a Nature Reviews Genetics article on sequencing-based approaches to study the microbiome that you might like to check out. For the AWIGEN 2 study, our sampling strategy was as follows. As Scott mentioned, we collected samples from 6 different sites in 4 different countries. The number of samples are indicated here on the y-axis, and the bars are colored by the different locations from which we collected samples. As you can see, the vast majority of samples were collected from Agincourt, or the largest number of samples, I should say, were collected from Agincourt, a semi-rural area in the eastern part of South Africa. But you can also see that we had quite a good number of samples collected from individuals living in quite different settings, ranging from Nairobi in a couple of urban slums, to Soweto, to Nanoro. At a very high level, once we collect these data, very basic questions should first be asked and answered, and I like to think of these as zeroth order questions. The first question that we typically ask ourselves in microbiome studies, as you know, is taking a look at things like prokaryotic diversity. So here we have prokaryotic diversity, it's the Simpson index on the y-axis, and we have the locations arrayed out on the x-axis. What I hope you can appreciate here is that prokaryotic varies across a rural-urban gradient, but not quite. Historically, of course, it has been demonstrated that rural areas harbor gut microbiomes that have much more diversity, for example, Nanoro, than very urban areas like Soweto. But it's interesting to note that places like Nairobi, which are technically urban, right? It's a very dense urban population. But remember, this is not a typical urban population. This is a slum dwelling population. We see a little bit of a difference in the trend that we observed before. We then decided to zoom in and take a look at what organisms or what bacterial genera varied most across sites. What I hope you can appreciate here in the top panel are a bunch of different common gut microbiome genera. Fissiocola, Bacteroides, Bifidobacterium, Prevotella, Cryptobacteroides, which is interesting in that it's an organism or a genus that is rarely seen in industrialized populations, but that we observe in our African populations. The same generally goes for Oribacterium. And then, of course, Treponema, a spirochete that has been well described to be associated with more rural lifestyles. Organisms like Fissiocola, Bacteroides, and Bifidobacterium seem to go up with urbanization. Interestingly, once again, I'll just call your attention to the far right of these plots. In red, you see Soweto. In gray, you see Nairobi. We see a little bit of a shift in that type of rural to urban variation that we typically experience, where we see much lower abundance of Fissiocola and Bacteroides in these slum-dwelling inhabitants. One thing to note about these individuals is that many of them were not born in these locations. They've traveled to there from villages that are outlying. By contrast, if you take a look at something like Bifidobacterium, which has been well demonstrated to be associated with consumption of dairy products, milk, yogurt, etc., you can see that here Nairobi and Soweto are quite similar. And this probably, we think, reflects dietary differences between those living in more urban areas and those living in more rural areas. I apologize for some of the formatting issues that arose here, but if you take a look over here on the bottom, you can see a heat map of the different strains or different species of these organisms that we observe. And I hope what you can observe is, for example, we see quite a lot of P. dorii and P. vulgatus in Soweto, almost none in Nanoro. For organisms like P. copri, we see them across the board. So, of course, taxonomy-based analyses are very useful, but what we know from previous work by many groups is that the gut microbiomes of individuals living in geographically diverse places can harbor very different taxa from one another, and also very different taxa from those organisms for which we have representatives in the reference database. For that reason, a lot of individuals in this space have started to do reference agnostic approaches or apply reference agnostic approaches to study the microbiome. In this case, what you do is you collect samples, extract DNA, sequence the reads, and then you assemble the reads into contigs or longer pieces of DNA that we can then group into draft genomes. These are called metagenome assembled genomes and enable us to really dig in to diverse strains and species that are present within samples. When we applied this type of analysis to the data that we collected, and just to remind you again, these are nearly 2,000 samples from 6 different sites. What I hope you can appreciate here on the far left is that we have quite a lot of different organisms that we were able to generate genomes for. Each line here in the tree represents a different organism for which we generated a MAG. On the inside track, you can see the phylum, and these are just color-coded. I hope you can appreciate that we ended up generating novel MAGs across all kinds of different phyla. And on the far outside track, you can see the sites. And so you can, I think, appreciate that we generated MAGs from many different sites, but you'll probably notice that the largest color representation that we have here is this kind of light brown color from Nanoro. And so Nanoro ended up being our site that generated among the most number of novel MAGs. You can see these novel mags separated by phylum over here on this histogram. And by far, we ended up with the largest number of novel mags in Bacillota A, which is followed by Actinomycetota. If you take a look over here in the top right corner, what I hope you can appreciate is that we have the number of bacterial mags that we generated by site. Those that have representatives in UHGG, a large representative database, are in dark gray. Those that are novel are in light gray. And as expected, in sites like Nanoro, the majority of mags that we generated were actually totally novel. But in something that might surprise many of us, we actually see novel mags arising from all kinds of different sites, including sites that we have heavily sampled in previous studies, such as Agincourt and Soweto. I'll call your attention finally to this small panel in the bottom right-hand corner. This is a phylogram or a phylogenetic tree of all of the different mags that we were able to generate that were novel for archaea. So while the vast majority of mags that we generate from these types of studies of the gut microbiome are bacteria, we were also able to generate novel archaeal mags. Now, antibiotic exposure is something that varies tremendously across sites, and we were curious to understand whether or not antibiotic exposure was associated with differences in taxa. For example, we know that organisms like spirochetes, in particular Treponema succinifasciens, are relatively abundant in Hadza hunter-gatherers. We also observe this in our more rural sites like Nanoro, where they practice subsistence agriculture, whereas they're quite rare in very urban places like Soweto. So we asked whether or not the proportion of individuals present in different sites had different antibiotic exposure based on the presence or the absence of Treponema succinifasciens. What I hope you can appreciate here is those who had Treponema succinifasciens were more likely to either never or more than a year, 3 years, 2 years ago have antibiotic exposure, as opposed to having more recent antibiotic exposure. This suggests that antibiotic exposure is inversely associated with the presence of Treponema succinifasciens, and antibiotic exposure might actually be one of the factors that's contributing to the loss of Treponema and many other spirochetes. If we take a look at the antibiotic resistance genes within spirochetes, here you can see the prevalence of those genes in different Treponema organisms, and these are different classes of genes. What I hope you can appreciate is that Treponema has almost no resistance to most of these antimicrobial resistance genes. Anything not present here was not present at all across the sites and across the strains. What you will notice here is that there are a few glycopeptide antibiotic resistance genes that are present in abundance, VAN-Y, VAN-H, and VAN-T. But I'll point out that these are not actually only antibiotic resistance genes. They're also genes that are important in the development of the cell wall. And so these are probably more used for typical cell wall production than for antibiotic resistance. In addition to this organism potentially being cleared by antibiotics, we also find that Treponema succinifasciens is inversely correlated with hip circumference. Here, once again, we separated the groups by individuals who had T succinifasciens present versus absent, and we looked at their hip circumference, which is plotted on the y-axis. What I hope you can appreciate is those who have the T succinifasciens present have smaller hip circumference. Now, this is not necessarily a causal relationship. This is an association, and this relationship may or may not be mediated directly. It might be mediated indirectly by other confounders. In addition to looking at bacteria, we also looked at the virome. Over here, you can see a heat map of each column representing a different viral genome that we were able to assemble from these samples. Everything with the blue tick here on the bottom represents a novel viral genome that we were able to assemble, and everything with the red tick represents a new Cras-like phage. What I hope you can appreciate is that across these sites, we were able to discover many novel viral genomes. Interestingly, when we take a look at viral richness over here in the right panel across the sites, we see pretty consistent richness across, with lower richness in Nairobi. Interestingly, we also observe a lower viral richness in Navrongo, and this is actually unexplained to date for us. Navrongo is also a fairly rural and horticultural site, although it is more industrialized than Nonoro, and Navrongo is based in Ghana. If we turn our attention back over to the left side, you can see the different viral genomes, and you can, I think, appreciate that we generate mostly novel viral genomes at each of these different locations, suggesting that while it is important to study diverse populations to learn more about the bacterial component of the microbiome, it's incredibly important to study it for the viral portion. If we take a look at classic, highly prevalent crassphages, such as P. crassphage, we can see that while we see that the Crassviralis family is prevalent across the sites, P. crassphage is actually relatively less prevalent. As we would expect, it is most prevalent in Soweto. In a final analysis, we decided to take a look at the gut microbiome and HIV. As you know, gut-associated lymphoid tissues can serve as a reservoir for HIV, and HIV can damage epithelial barrier integrity, allowing microbial translocation that in turn promotes chronic inflammation and immune activation. We also know that antiretroviral therapy is not sufficient to control HIV-related comorbidities or return a healthy microbiome composition. And so what we sought to do was try and understand the microbiome and its relationship to HIV in these sites. Now, only a few of our sites had adequate numbers of people living with HIV and data on this in order to do this analysis. The site at Bushbuck Ridge, which is also known as Agincourt, Soweto, and Nairobi. As you can see, these are the number of samples from people with HIV, without HIV in gray, and those living with HIV in blue. What I hope you can appreciate is if we take a look at the combined bacterial richness across these sites, we see a higher richness in people living without HIV versus those living with HIV. And this is statistically significant in 2 of our sites where we had large numbers of individuals living with people living with HIV, Agincourt or Bushbuck Ridge and Soweto. Finally, we were able to take a look to see what species were either enriched in people living with HIV or depleted in those living with HIV. Those taxa are represented or listed here. What I'll note is that these are actually in some ways common with microbiome signatures derived from Western cohorts, but in many ways, many of these organisms are quite unique to these African populations. We then created a machine learning model based on these data to create a microbiome signature and then applied this microbiome signature to see if we could predict individuals who would be living with HIV. As you can see, data that went into this came from Agincourt, Soweto, and Nairobi. So we tested on those samples that were left out, Nonoro, Navrongo, and Damamo. We don't have information on HIV status from Nonoro and Navrongo, but based on population statistics, we don't expect it to be that high. If we take a look at Damamo, we do have information on this. And what I hope you can appreciate is that among those people living with HIV in the blue, we were able to predict a large proportion of them as positive, about 0.7 as the fraction. Whereas of those living without HIV, our test was very accurate in predicting that they didn't have HIV. So in conclusion, for this section, AWI-Gen2 is a large cross-sectional gut microbiome study from 6 communities in 4 countries. Microbiome composition does not reflect a linear gradient with population density going from very rural to very urban, but is influenced by the complex interplay of lifestyle factors, as we saw for Nairobi. African microbiomes contain huge amounts of previously undescribed bacteria, archaea, and viruses. And we hope that this study population will enable the discovery and application of this African-specific HIV microbiome signature. As Scott alluded to, we're just scratching the surface, and there is a lot more analysis to do. I hope we'll have the chance to work with those of you on the call at some point in the future. And with that, I'll turn it back over to Scott.
Thanks, Ami, for that presentation of the work. And I'm going to just conclude with a few remarks about the relationship of our teams with each other and with our stakeholders. And I think of this as an engine for sustainable research. So it's what makes this research possible in the long run. So there are two motivations for this. The one is that we really would like to do things ethically and correctly, but we're also designing this as a longitudinal study. We'd like to go back in 2 or 3 years' time to do a 5-year follow-up. We'd like to go back in 10 and 15 years' time to follow up the same individuals and the communities. And we're only going to be able to do that if these different stakeholders feel that there's an authentic relationship that we have with them and that they're getting something from what's happening. So the three stakeholders are the local research infrastructures, the excellent scientists that we work with at the various sites, the communities in which they work, and then the participants within those communities who gave samples and their time as well. So the first is the local research infrastructures. I think that goes very much to the question about helicopter science that someone asked in the chat. I think it's critical that these infrastructures are involved and capacitated. First of all, through the entire cycle of the research. They need to be involved from the beginning when we plan the project and we come up with the hypotheses and the scientific questions, all the way through to the end in terms of appropriate acknowledgement and authorship and opportunity for authorship. There's also opportunity for training, not in the field work where they are the experts, but in the microbiome analysis and the bioinformatics analysis as well, to make sure they have opportunity for that. Not always easy for them because they're very busy. And the fourth part I want to mention, which I think is very important, is ensuring that they have we can support them with getting core funding. Many of these research sites are funded project to project, and that makes them very vulnerable to short-term changes in funding. So to really capacitate them, they need to start building up core funding where they can explore new ideas as well. Then community engagement. We are doing these projects as part of communities. They're not just the participants. People are living in the community, and we need to get the community to support the work that we do. And there's various reasons for doing this. We need their support practically. I think also by understanding the community better, we're more likely to be asking the interesting and the hard questions that are appropriate. So yes, we need to understand if there are any sensitivities about the research, but we need to understand where the community is at. And the particular people who we found to be very important are the field workers, the specific field workers who work in our project, because they're not just employees going out doing things. They are the ambassadors for our project. If they are bought into the project, if they are first-class citizens in our project, they're going to do a much better job, and we're going to learn from them. And the final part, I think, is making sure that we give the community as a whole feedback from the research that we're doing. I think there's opportunity to make an impact in health. And then the final part, which is newer work for us, and we're finding quite difficult, is participant feedback. We have started a pilot project at 3 of our sites where we're going back now to the participants to give them personalized feedback from our research results. And the first thing I want to say about this, the preliminary results we've had, the preliminary response from our participants, is that the participants really, really, really want feedback, and they appreciate this individualized feedback. And this goes both to the ethical imperative, I think, of making sure that people that we work with get something from the work that we do, but also from our self-interest point of view, we now know that if we want these people to participate in future studies, we need to look at how we can give them feedback. And this comes then to what sort of feedback can we give them? The other point, I think, is that this feedback is also an opportunity for us to do outreach into the community for people in the community to understand the work. Here's a photograph of one of our participants. She's got her personalized feedback on her lap. She's looking at a cardboard-based microscope with some prepared slides with microbiome, with bacteria on them. And the field worker is using this as a way of explaining what the microbiome is, what impact the microbiome has on health, and what bacteria are. So I think we can use this as an opportunity to explain things that we wouldn't be able to otherwise. So the question then comes to, what feedback do we give? Now, up to now, we've always given feedback at the point where we collect samples and data from participants. And these are generally not microbiome related. So for example, if we take blood pressure, or if we do a glucose test, or we do an HIV test, we will give feedback to participants, and we will give them a referral to the clinic for treatment or for counseling where appropriate. Here we're talking about giving individualized results about the microbiome, and what results do we give back that are both scientifically valid and meaningful to the participants. And we need to bear in mind where our participants are, and I've got here on the right-hand side, there's a pie chart of the educational level of the AWIGEN participants. And then in another project that we're doing with the South African Research Organization, the CSIR, we're doing a community mapping project in Johannesburg and Pretoria. There's a very different educational profile. So when we're giving this feedback, we need to understand where our participants are coming from. But I think there's also an ethical responsibility that we are not being paternalistic and patronizing, and we make a real attempt to give feedback that is useful to our participants. I want to emphasize that we make it clear to the participants that the feedback that we're giving, the personalized feedback that we're giving, is not actionable in the same way that a blood pressure test or an HIV test might be actionable. So at this stage, it's just for their interest. And we explain that the purpose of this is primarily for them to be helping us to understand what the microbiome is doing, but we still find it useful and important to give them back their results. So what we're doing in the study is we have an in-person meeting where field workers or scientists explain what the microbiome is, remind them what the study was. We tell them the importance of the microbiome, what they can do in their community to improve their microbiome status. We explain what the personalized report is telling them, and then they get their personalized report back. And then there's someone there who can answer their questions if they have individual questions. So we're testing now at the different sites how to do this. Do we do this with the individual visits to the household, or do we do it within a group situation? We're looking at what goes in that personalized report. This is an example of a 2-page report that participants in Bushbuck Ridge got. So this is in Shangaan, which is one of the local languages. There's a brief description of the microbiome, the Switsongwana, what the microbiome is and why it's important in health. We tell them the number of bacterial species we found in their gut and how it compared to the rest of the community. And we explain to them at this stage, we're not saying that's good or bad. It's just a fact that they may be interested in. And then there's something about the microbiome diversity in their gut compared to their community and the other communities in which we're working. So I think that gives you some flavor of the feedback that we're giving. So this is work that should be planned from the beginning. We didn't plan to do this from the beginning. We planned only on some group feedback initially. It would certainly have been better if we planned it all from the beginning. There is a significant cost factor. At the first site we did this, I estimate that the individual cost, when you try and give feedback to 500 participants, where we've got an existing platform, is about $22 per person. So that's a fair amount of money. Although I think if you look at it in the overall cost of a project, that's not a huge amount of money compared to, say, doing DNA extraction and sequencing, or even the cost of a fecal collection kit. And we're very fortunate that the NIH were, I think, a far-sighted funder and have supported our pilot project to see whether this is something that's worth doing. So with that, I'm going to conclude and we'll take questions. There were lots of people involved in this project, the participants, our community advisory groups, the field workers, and the broader AWIGEN team. The AWIGEN team at the different sites, at the Sidney Brenner Institute, the different field sites, are really amazing people that we worked with, and we're very grateful to them. The core microbiome team, the AWIGEN microbiome team worked on this. Fantastic collaboration between Stanford and AWIGEN, and I think, as Volker said, a model for how this type of work should be done. Our various funders were very generous and were very open in the funding. And if you are interested in finding out more, we do have a preprint that you can look up, Expanding the African Gut Microbiome, and there's a QR code that you can scan. So Volker, I think I'll stop, and Ami and I will take questions and listen to any comments and feedback that we get.
Thank you so much, Scott and Ami, for the fantastic presentation. I really, really like the fact that you not only talked about the project, but everything else that sometimes gets ignored with, carrying out research projects, and just giving like a bit of a, what we could have done differently and what might have worked better, and just like how collaboration works, and just, we want the results, but we also want to know what's happening with the participants as well, and how they feel about the research being done in their communities. I think this is just a fantastic example. So I could say many things, but there are actually quite a few comments and chats in the chat box. I've lost my chat for a bit, but just a second. I will get back to the first set of questions in a little bit here. So I think of an easy one.
Sorry, I should just maybe just have I should say that maybe the last part of my talk made it sound like we had everything planned from the beginning and we knew exactly what to do. Certainly wasn't the case. We had some ideas, but we made some mistakes and we omitted some things. So this has been a learning experience as well. So I don't want to set up what we did as the ideal way of doing things. Certainly not the case.
No, thanks, Scott. Thank you, Scott. I think that came across. So the first question is very simple, I think, an easy one. The question was from Frank Onyambo. Is the microbiome data for AWIGEN available for researchers in Africa?
So the answer is it will be when we've got a paper under review, and certainly once the paper, if the paper is accepted, then the data will be made available. I just want to talk about that, is the data availability is obviously very important. In H3Africa, there was a very intense debate about making data availability, but also giving the scientists who worked on the project an opportunity to have a reasonable first cut at the data. But our data, we hope, will be available very soon.
Thank you, Scott. The other question, and Ami, please feel free to chime in. The other question is, I'm sure you can read this in your chats as well. This is from Ruari Robertson. Did your prediction models adjust for cotrimazole, I hope I pronounced that correctly, used in people living with HIV?
Thanks for the question. So no, we did not adjust for clotrimazole use in this particular model.
Cotrimazole, got the pronunciation. All right, cool. And the other question was from Gilders. He's asking, which machine learning algorithm did you use for the prediction, and how much data did you have to train versus validate the models?
Thanks for the question, Gilda. So we used all of the data from the 3 sites where HIV was quite prevalent. So this was the Agincourt site, Soweto, and Nairobi to train the model. We then took that model and we validated it on an external site, which was Demamo. We also tested the model on the 2 other sites where we don't actually have data on whether or not people have HIV. That's Nanoro and Navrongo. There, the model predicted that HIV prevalence was very low, which is consistent with population prevalence information that we have. But we can't directly address the accuracy of the model in those 2 sites, but we did validate the model in Damamo. Thank you for the question. And in terms of the numbers, it was, in the around 100 something people living with HIV, as I recall.
Thank you, Ami. And feel free to ask follow-up questions in the chat if you'd like to. We have plenty of time. Another question for, I think both of you is from Heather Jaspin. She says, thank you. Have you looked at the virome in people living with HIV? Do you know whether the PPTs were on, do you know whether the PPTs were on ARVs? I'm not quite sure, but you probably understand the question, so.
Yeah, I'm happy to take that one. So that is a great question, Heather. And we're very excited to look at the virome in people living with HIV. This is on our list to do next. I think it's a really, really pertinent question and super exciting to evaluate, especially since some of the antiretrovirals that these individuals were on might affect, for example, reverse transcriptase diversity generating retroelements, et cetera, that might be in the microbes or the viruses within the gut microbiome. I will say that I think it would also be very interesting to generate RNA-seq data so that we could take a look at RNA viruses in the gut virome, but we haven't done that to date. And there is one small caveat in that we collected samples in these OmniGut DNA genotek tubes that are rated to protect against DNA degradation, but not RNA degradation. We've done some pilot work to try and assess whether or not we can get good transcriptomic data from samples preserved in this way. Dylan McGeaney and Mai Dvorak in our group led a study that was published in Nature Biotechnology on that. So we think we might be able to, but it's still underway. And to your question about ARVs, yes, we have information about whether or not people were on ARVs and what ARVs they were on. I'll just note that most of these samples were collected a handful of years ago. And so the antiretrovirals that the patients were on or the individuals were on in this cohort are not the most modern antiretrovirals, for example, DTG. So it'll be important to repeat this type of study in the future with more modern antiretrovirals as well.
Hey, thank you so much, Ami. I think there's another question for Ami specifically from Sumaya Kwidi. How do you think we could expand this microbiome atlas across the entire continent?
I think that's a question for you all. I'm glad to hear that someone's asking the question because it sounds like they think that it should be done. And I think, Sumaya, it is a really important part of exploration. I think it's so interesting to me how different the microbiomes are of individuals living in sites that have pretty similar lifestyles. For example, Nanoro and Navrongo. By the numbers, population density numbers, et cetera, they look very similar, but their microbiomes are very different, suggesting that there's a huge amount of lifestyle variability and exposure variability across these sites. So I think what it's going to take is a lot of grassroots champions. I encourage people to reach out to me, Scott, Voke, and others to bring to us your ideas. And of course, there's also the practical necessity of resources to enable us to do this as a community in the right way. I did want to also mention, I think I forgot to respond to Gildas's question before about the machine learning models that we used. We used a lasso model, and details of it are in the preprint if you'd like to check it out.
If I can just add to Ami's answers, I think obviously a project like this requires money from a funder like NIH or the Wellcome Trust or some other funder. It's not cheap to do this work. But I think there's some appetite for that. But the most important part is there has to be partners on the ground who can do the work. We would not have been able to do this work if it hadn't been for the health and demographic surveillance sites at the various sites, working with the scientists who've invested in some cases over decades in the platforms that they have. So I think that's how this work is going to really take off. Because the other thing is we don't just want to go out and sequence the microbiome. We want to tie this to phenotype, to health, so that we can understand what's happening in the microbiome.
Thank you very much, Scott and Amy. I think also that there's been lots of, I mean, I know I'm biased, but there's lots of compliments as well for the amazing work being done. And I think that's encouragement to, like, you know, to advance towards more frontiers in this field and just go on from there. I think there's a question. I don't think that's necessarily for you, but I mean, anybody feel free to answer the question in the chat about funding for wastewater-based epidemiology initiatives. Feel free to answer that question from Grasa Kandada. There's a question for Scott from Joanne in the chat. When the feedback about the microbiome was shared with participants, what was the response? I know Joanne has also commented previously that she's very happy and excited about this people-based approach. So that's a question for you, Scott.
So we are busy working on this at the moment. We have finished at one site. We're halfway through the second site, and we'll be doing the third site next month or the end of this month. So we haven't completed the analysis of the results we've got from the participants. I've read through all the comments from the participants at the one site, and generally people were very positive about it. I think there's a lot that we have to learn still about what's the best way of explaining things. But I think the bottom line is they really, really appreciated it. I mean, that comes through, is they say that again and again, is they appreciate us coming back, and because not all projects do this. And for us, we're wanting to go back in a few years' time. I think it's critical that we invest in this.
Thank you, Scott. I think something that comes out there is that for the continuity we hope to see in microbiome studies, the longitudinal studies we hope to accomplish, we need to make sure we have these things in place so that the participants are receptive to, you know, further, you know, studies in their communities, because we need this guidance.
Yeah, I should say it's a bit easier for us in the sense that we're working with the HDSSs. I think if you are doing a study where you are advertising for volunteers and people join in, it's maybe not as easy to go back to participants in a year later or 2 years later to give them feedback. Because we're working with HDSs who have a relationship with their communities, it's easier for us to get back to the communities and to get back to the participants and to get feedback from them.
Thank you, Scott. I think John has a few more questions in the chat. So I think one thing that might be helpful in answering those questions as well is, you know, originally Abidjan was a genomics project, but somehow we're able to incorporate the microbiome aspect of it. So our question now is, are you included in any qualitative studies? And she's trying to get all participants in every study to be microbiome advocates and enthusiasts. I don't know if you have Amy, feel free to chime in as well. So.
Yeah, I mean, this is a great question. I do think that it is fascinating to see how interested people are in their microbiome. Like, even when we can't really offer anything very substantive in terms of, you know, cause and effect relationships between their microbes and their health, or what they do and their microbiomes, I think it is a touch point that's just intrinsically fascinating for individuals. And so I do think that thinking about ways that we can encourage our participants to become enthusiasts and advocates for this type of work is important. To the qualitative studies question, perhaps I'll turn over to Scott.
Yes, so this is, as I said in my talk, this is something that would have been much better if we planned it from the beginning. So it was planned towards the end of the project when we had practical issues about deadlines, when money had to be spent, and how much work we could do in the field. So we will be doing some qualitative work with it, but not as much as we would like to do it. We see it as an initial study. And I think I should also give credit to Dylan McGeaney, who's been pushing and leading this aspect of the work. We do hope to have a paper on this.
So maybe I'll just say it's been a real honor to participate in this research. I have learned so much from Voke, Scott, the many, many colleagues who were listed, the many colleagues who we can't even fit onto a slide because it took a huge number of people to bring this work forward. I want to highlight that I think we all learned a lot about health and demographic surveillance research and how to participate with communities to help design research. And I think that this has made me a better microbiome researcher and a better scientist and person. And so I'm very grateful for the opportunity. I think there is a lot of opportunity to grow as people and scientists if we engage in this type of collaboration. So it's been very fulfilling. And a big public thank you to Scott, Voke, and many others.
It's been a great collaboration.
Thank you so much, Ami and Scott. Thank you so much for the incredible presentation, for your time. Sorry, I had a bit of a technical difficulty there for a second. Hopefully I'm okay now. I can't put on my video for some reason because I think I rejoined, so I don't have the host ability. So please, my excuses. But thank you so much, everyone, again for your attendance, for the amazing participation. Thank you so much for the time. I think this has been fascinating. This has been amazing. And just before, we're putting this out here, so you feel free to scan and sign up for the African Microbiome Consortium. Just let us know what projects you're working on so we can coordinate better. We do have our African Microbiome Special Interest Group meetings every third Monday of every second month. We'll be having a meeting. The next meeting is on the 21st of October at 4 p.m. Central African time. Please feel free to join in. Reach out to me or to Tino or to Francis for any information in this regard. We'll be happy to send you the links and the calendar invites for these meetings. And just again to say thank you so much. We're going to be sending out.
Sorry,
please go ahead.
I just wanted to say something before you stop.
Yes, please go ahead.
There was a request for a group photograph.
Yes, there was by Rim Salam, and I did ask if it was going to be possible. Tino, is that going to be possible for a group photograph? So that would mean that everybody would have to switch on their cameras as well. But before we do that, just to say that thank you so much to the organizing team. It was a long collaborative effort, a team effort. Thank you for your time. Thank you for the input. Thank you so much to the H3Africa Coordinating Center, especially to Dr. Francis Agama, I should say that, Dr. Francis Agama, and to Tino. Tino's been amazing. Thank you so much to everybody that submitted abstracts. Sorry to those we couldn't quite present your work today. We look forward to hearing more updates on your work as we proceed in this process. And we look forward to having that consortium at some point where we can give you some good feedback at the next African Microbiome Day. And with that said, I don't know if Tino could probably just share the slide for the African Microbiome Institute symposium coming up in November. The African Microbiome Institute also has, they're also a part of the African Microbiome Special Interest Group. Like we said, it's not a one lab initiative, it's several efforts. And the African Microbiome Institute, they're having a symposium coming up. Please feel free to get involved in the symposium as well. And Joanne also has something coming up on the 1st to the 3rd, I believe, of October on the Vaginal Microbiome Consortium. Please feel free to reach out to her to get more information on that. A bunch of these were already shared in the chat a bit earlier, but please feel free to reach to these people for more information. Would like to take a picture if it's possible. My background is, great, I guess. But let's see if we could please turn on our cameras so we could just grab a picture of everyone in attendance. That would be great. Thank you so much, everyone. I think I have to stop speaking, right? So that I'm not overlapping anybody. Let me stop speaking. Tina, let us know when you're ready.
Okay, I've been taking as people turn on.
Okay, thank you so much, everyone. We're going to be sending feedback forms out, and you're going to get like a participant a certificate of participation or attendance, whatever is applicable to you. Please let us know what you think. We're very, very excited about feedback and would like to do better and would like to improve on this. So thank you so much, everyone.